All right, everyone, we're just about to get started, so please find your way to your seat. Okay, well, hello. My name is Talmo Pereira. I am a faculty member at the Salk Institute, and it is my pleasure to serve as the tutorial co-chair as part of the Cosyne organizing committee. And in this capacity, I am thrilled to welcome our main Cosyne tutorial speaker for this year, Alex Williams. Very brief intro on Alex. Alex is an assistant professor at NYU's Center for Neuroscience and a project leader at the Flatiron Institute's Center for Computational Neuroscience. He is a 2025 McKnight Scholar, DOE Computational Science Graduate Fellow, Goldwater Scholar, and I'm sure many honorifics from high school and middle school as well. Good job, Claude. He did his PhD with Surya Ganguly and a postdoc with Scott Linderman at Stanford. More importantly is he spent some time at the Salk Institute working with Terry Sinofsky before making the mistake of going to the wrong coast for his faculty position. His lab developed statistical methods for understanding neuropopulation dynamics at the single trial level, tensor decomposition, time warping models, and more recent work on things like optimal transport and shape metrics for comparing representations across conditions, individuals, and species, and as I hope we'll see, even across models and biological neural networks. So join me in welcoming Alex Williams.
Thanks, Talmo. I can't believe Claude missed my National Honor Society as well. I did so much community service for that. Okay, thanks everybody for coming out to this tutorial and for your interest in hearing about quantitative methods for comparing neural population codes and high-dimensional neural dynamics. So I'll just jump right in and start by giving some motivation for this line of work. And the way I really think about it is, if you look at the broader field of biology and ask what works, how do biologists understand complicated systems, I think you'll notice that in many cases they lean very heavily on a so-called comparative paradigm or comparative lens. And this is really built into the foundations of biology going back to Darwin, and even before Darwin, people were very interested in understanding what are the similarities and differences across different animal species, and how do those similarities and differences map onto advantages in evolutionary fitness and adaptation. And in modern times, this has led to entire subfields that are explicitly focused on this idea of drawing comparisons between different biological systems, whether it's at the scale of anatomy or lower level cellular biology, or even interacting animals in ecological contexts. So these are just 3 example textbooks that summarize entire fields of study on this perspective. And you can also find peer-reviewed journals that are publishing new papers each and every month that are, again, explicitly focused on this idea of comparative analysis. So I couldn't help myself but give a little bit of a digression on how comparative analysis is helpful to biologists by giving you the specific example of how we came to understand how kidneys work. And I'll keep this at a very high level, but I think it's a really interesting history here, that if you look at the history of how biologists studied kidneys and came to understand how they work, a really early clue about how kidneys worked came from an explicitly comparative analysis where people went out and looked at the structure of kidneys in desert animals like a kangaroo rat and compared the structure of their kidneys to animals like beavers that live in aquatic environments with plentiful fresh water. And, oversimplifying here a little bit, the main difference that you see between kidneys in these two kinds of animals is that there's a big difference in how much the inner region of the kidney, called the medulla, the size of that region relative to the outer layer of kidney, which is called the cortex. And this led to an explicit mechanistic hypothesis for how kidneys worked, which is, again, oversimplifying just a little bit, that your tubules carrying fluid go from the outside into the inner core of the kidney, and there's a high concentration of extra tubular salt in the inner kidney, which pulls water out of the tubule and then concentrates the urine. And if you look across animal species, you can actually draw a pretty tight correlation between how much area there is in this inner region of the kidney versus the outer region, and how effective the kidney is at producing a very, very concentrated urine and preserving water. So this is, I think, one of the cleanest examples of how comparative analysis, at least historically, led to a very specific mechanistic hypothesis for how a biological system functioned, and then that was later tested with direct experiments successfully. So I think that's a bit of cool history, and it's not going to be as simple when thinking about the brain, but my goal and dream would be that we could leverage a similar sort of approach in understanding neural circuits and how large populations of neurons conspire to produce intelligent and adaptive behaviors. So what I'm going to sort of pitch you today is the idea that we can start developing a comparative analysis of systems level neuroscience. And there's many forms that comparative analysis could take. So for example, we might record hundreds of neurons in motor cortex across 2 different animal species performing similar reaching tasks or forelimb movement tasks. And then we would be interested in asking the question, to what extent are the neural population dynamics in motor cortex similar or different across these 2 species? What are the aspects of the computations that are similar, and what are the aspects that are different? Another form of comparative analysis would be, even within potentially the same individual, you might simultaneously record neural activity in 2 different brain regions. So for example, maybe in a visually guided decision-making task, you might record from a prefrontal cortex, which is thought to control executive functions related to decision-making, and at the same time record in a visual area like V4. So we think both of these brain areas are maybe contributing to the task at hand, but we would hypothesize that they're formatting information differently or representing the task information in different ways. So again, this would be a comparative analysis where we record hundreds of neurons across 2 or more brain regions. And again, the question is, what is similar or what is different about those different population level neural representations? And then finally, we can also make comparisons across individuals of the same species. So in this example, maybe you record neural population activity across 20 different mice, all performing the same task. And I'd argue that as a field, we have very little understanding currently about what is the natural level of animal-to-animal variability at the level of neural population dynamics and representations. And the reason for that is not too hard to, if you think about it for a minute. It's only been within the last 10 or so years that we've had cheap and scalable and easy to use and deploy methods for recording from enough neurons from individual animals so that we can repeat this experiment multiple times and gather up enough statistical power to say something with confidence about animal-to-animal variability and not just attribute that animal-to-animal variability to measurement area. So I think it's a really exciting time. I would point to initiatives like, for example, the International Brain Lab, which really took this to the extreme where they distributed the labor across many different experimental labs and recorded over 100 mice, all performing the same task. And they made recordings in different brain regions, but nevertheless, there's still a lot of data there that we can maybe make apples-to-apples comparisons across different animals and finally say something, I think, rigorous about animal-to-animal variability in neural circuits. Okay, so that's a high-level overview of what I want to cover today and the kind of vision that I'm laying out with this sequence of methods that I'll tell you about. And this is the structure of the tutorial. Basically, I've broken it in roughly into 4 parts with short breaks between part 1 and 2 and a short break between part 3 and 4. And then there's a larger break in the middle where people can get coffee. We have some Jupyter notebooks that you can play around with. So if you want to get some sense of how you use these methods when you go back to do research in your labs, there's time for that there. And even though I have set aside some time for explicit Q&A. I think it would actually be optimal if we could get, as I go through the material, questions from the audience as I go. So we have a number of TAs in the room. I think this is maybe I don't know, I have the picture of TAs in a few slides. But we have TAs in the room who will pick up the microphone and distribute it throughout the room. So please just raise your hand if you have a question and want to interrupt at any time. I mean, we're going to be here for 4 hours about, so it's going to be more fun for me and probably for everybody if we make this as interactive as possible. Okay, so what am I trying to tell you in this tutorial? At a high level, the goal of this tutorial is to expose you to a variety of different comparison methods or quantitative methods for comparing neural population representations and dynamics. And the spoiler alert is that there's many different ways to do this, and that's why we have 4 hours to cover all of this ground. And because there's so many different ways to think about this problem of comparing neural circuits, when coming up with the material for this tutorial, I made a conscious decision that I wanted to focus conceptually on what these different metrics are actually measuring about neural systems, and have a discussion about why or motivate why that may or may not be a good thing to quantify in neural population dynamics. And because of that, there's a little bit less emphasis on how you run these analysis on your laptop or on your computer. And that's by design. The first thing that I'll say, and this will hopefully become clear as I go through the specific material in this tutorial, is that to actually run these things on your data is pretty simple. A lot of the routines that I'll talk about are actually built into scientific libraries like in MATLAB or in Python. So there's not a lot of effort that you need to go to implement these methods from scratch. There's a lot of interesting mathematical details that I'm also going to hide under the rug. But again, those are mentioned in our Jupyter notebooks and also in the papers that I'm going to be citing throughout. So I'm going to try to keep most of the content or of the talk at a higher conceptual level and give you intuition about all of these methods without going too much into the weeds about mathematical details or how to implement these in code. Okay? But there is a link. We have a number of really nice step-by-step tutorial notebooks. I'll leave that up for a second in case anyone wants to take a picture of that link or go there on their laptop now. And that will walk you through some of the more step-by-step specifics of how you run these analyses. Okay. And again, yeah, I want to give a big thank you to the TAs for this tutorial. The TAs really went through all the notebooks and made sure that they're polished and ready for you to work through. So that was a really, really big help in pulling this together. I want to explicitly give a thank you to Sarah Jo Venditto, who was the sort of lead TA and helped not only do all of that of polishing the notebooks, but organize the TAs into a coherent force behind this. So thanks, Sarah Jo. Okay, and yes, please raise your hand if you have any questions. And the TAs are, I've been assured the TAs are near the microphones and ready to bring them around the room as necessary. Okay. So let me dive into the first part of the tutorial, which is going to go over some example applications to give you a flavor of what you can do with these methods, and then also build up some fundamental ideas that are going to come up again and again over the course of the tutorial. Okay, so for the first example analysis I want to show you is motivated by comparing representations across different brain regions. So we looked at a data set that was released by the Allen Brain Institute. This is the Allen Brain Observatory. These are mice recorded with neuropixel probes, passively viewing a visual scene. So it's a 53-second movie clip from Orson Welles' Touch of Evil. And The first thing we wanted to look at was just what are the similarities and differences in neural population responses across brain regions. So for each brain region, the data looks something like what's on the left of the screen now. So you have some trial averaged neural firing rates over time. So each row of this matrix is a time series, a continuous trace that represents the average activity of that neuron when seeing this particular movie. And something that's interesting about this brain region that's on the screen is it's an area in auditory cortex, which is not really a canonically viewed as a visually responsive area. But you can perhaps see, and I don't know how visible my cursor is, but if you look carefully, you can see these vertical striations on the time series, and those vertical striations are indicative of neurons responding to an interesting event that happened in the movie at those times. So there are visual responses in auditory cortex, and you could imagine that you could do principal components analysis on this matrix. You could do other sorts of manifold analysis to analyze or analyze the information content of this time series. And you could have an entire poster, lecture, paper on what does auditory cortex care about in this visual scene. So that would be a very interesting question and in many ways like a conventional sort of analysis in neuroscience. And what we're going to instead try to do, instead of just doing a deep dive into one of these areas, we want to take a bigger picture view of the full data set, which looks like this. So now these are all 55 brain regions that have at least 50 neurons recorded in the data set. And each one of these brain regions has all of that structure that was present in the first example that I was showing you. So each brain region is a number of neurons by number of time bins matrix, which is a high-dimensional time series. And we'd like to understand, what are the patterns in this data set that I'm showing you here. So this is my best attempt to show you the data in its raw format. And hopefully you can appreciate that this is a really complicated data set, and it's really hard to look at the raw data and make sense of it directly. So the leap of faith that we're going to make today is that it might be possible to boil down complicated data sets like this into something simpler. And in particular, we're going to think about boiling this down into a matrix of distance comparison. So it's the raw data is high-dimensional time series from 55 brain regions. We're going to try to boil it down to a 55 by 55 matrix, where each element of this matrix is somehow going to summarize the distance between brain region I and brain region J. So this is going to be a summary statistic that is low if brain region I and brain region J have similar dynamics going on, and it's going to be large to the extent that those brain regions are representing the video differently. Okay? And the entire tutorial is going to be focused on how do you come up with good distance measures that create a distance matrix like this. Okay, but for now, let's just say that we can get such a distance matrix. What are the interesting things that we can do with it? Well, one thing we can do is we can actually take this distance matrix and hand it off to off-the-shelf routines for data visualization. So what we did in this example was we took the distance matrix and we passed it off to what's an algorithm called multidimensional scaling. It's a really classical and well-known algorithm. What multidimensional scaling does is try to find a low-dimensional set of points that capture the pairwise distances in this distance matrix. So if we do multidimensional scaling and then followed by principal components analysis to push it down to 2 dimensions, we get a picture that looks like this. So now each point in this low-dimensional space corresponds to a different brain region. So there's 55 brain regions and therefore 55 points. And each pair of points, if 2 points are close together, that is indicative of their distance being small, i.e. they're expressing similar information about the task. And if 2 points are far apart from each other, that correlates with them being somehow dissimilar from each other in how they're representing this visual task. And the neat thing that pops out of this analysis is that it's in some sense a fully unsupervised analysis. We're just trying to take this 55 by 55 distance matrix and find a low-dimensional set of points to visualize the data. This algorithm knows nothing about the anatomical relationships among these brain regions. The distance matrix is built just on the neural firing rate responses. And we get this low-dimensional projection, and if you color the points in that low-dimensional space by the colors in the anatomical Allen Brain Atlas, you see that the cortical areas in teal all sort of clustered together, in this case on the lower right-hand corner of this projection. The pink regions are thalamic regions that are also visually responsible. This is mostly visual thalamus regions. And then this final cluster of points are mostly visually non-responsive regions in this task, and these are mostly in the hippocampus. Okay, so I would argue that this is a very non-trivial result, which is showing that the functional similarity of neural population dynamics actually reflects the anatomical similarity of these different brain regions. And this is true not just at the gross level of cortex versus thalamus, but if you zoom in on smaller parts of this projection, you'll see that, for example, all layers of the primary visual cortex cluster together, layers of the rostrolateral visual cortex cluster together, and this is also true of different divisions of cortex, like auditory cortex versus visual cortex. So there's a lot of interesting and non-trivial structure that pops out of this analysis. Okay? I want to give you one other example before moving on, and this is going to be an example looking at animal-to-animal variability rather than variability across different brain regions. So this is looking at animal-to-animal variability in the head direction system. Specifically, these are recordings in the post-subiculum of mice. This is data released by Duskowitz et al. This is coming out of Adrian Peyrache's lab. And the really cool thing about this data set is that they have data from 30 mice. So this is actually a lot of data that we might be able to say something rigorous about animal-to-animal variability in this system. So just to give you a sense again of the raw data, we did principal components analysis individually for each mouse. So now we have 30 PCA plots shown here on the right side of the screen. But we're doing this on the head direction tuning curves. So the head direction tuning curves are these sort of bumps of activity that are in response to a circular variable, a periodic variable of the animal's head direction. So because this is a one-dimensional periodic variable, the neural firing rates are tracing out a one-dimensional curve in firing rate space, and this is on a closed ring because, again, the underlying variable is periodic. Okay, so it's not it is expected that you get, when you run PCA, you get ring-like manifolds in each animal. But if you look from animal to animal, you can see a lot of variability in the shape of these rings. So we were wondering whether this is indicative of true variability from animal to animal, or is this somehow just experimental noise? So we did a similar analysis to what I just walked you through on the previous example, where we built up a 30 by 30 distance matrix that summarized the distances between all these representations, and then did multidimensional scaling in PCA to visualize the data. And what you find is that the top principal component of animal-to-animal variability in this data set seems to be correlated with the average width of the tuning curve. So there actually does seem to be some interpretable animal-to-animal variability. And you can validate that. I'm not going to go into the details of this, but you can imagine doing cross-validation procedures where you, for example, split the neurons from each animal into two populations and then measure the distance of an animal to itself versus the distance of an animal to one of the other animals in the data set. And what you find is that there's a significant difference, that the distance of an animal to itself is lower, that's what's shown in this blue histogram, than on average what you see when you measure the distance across animals. So we think that this is a true signature that there's real animal-to-animal variability in these data sets, and it's something that, to our knowledge, hasn't really been studied carefully before. So this is maybe opening the door on research into that direction. Okay, so for more details on, oh yeah, sure. TAs, stand up and the microphones are still in the stands. I want those. Yeah, there we go.
All right, thank you. So you said this might be indicative of true animal-to-animal variability.
Yep.
In contrast, maybe to measurement error, but
well, yeah,
so
I would be a little bit careful there, but
because when you talk about tuning width, it could also be that depending on where you measure in the head direction system, there might be different tuning width, and this is just reflecting where exactly these cells were recorded.
Yes,
so
yes.
How do you
that is the caveat I was brushing under the rug. That yes, okay. there could be. You would have to. It's difficult to control all the variables of that experiment, including like the probe position and things like this. I do think that it's not fully explained by that, but it might be difficult for me to prove that to you today.
Okay,
yeah.
Thanks.
Yeah.
I really like your shirt, by the way.
Okay, thank you. Thanks for the question. Yeah, please keep the questions coming. Yeah, somebody else over there. Okay. Okay, so those are just two quick examples to give you a taste of the kind of analysis you can do. You can read more about them in this preprint that was led by João Barbosa, who's also here at Cosyne. I'm sure he'd be happy to talk to people about those results. Okay, so the core idea that I just kind of introduced by way of example is that we might be able to make systematic comparisons across different animals or different brain regions by defining a distance function, or what mathematicians would actually call a metric space over neural representations. So just to summarize what I showed you by way of example, is we had data sets that looked like this, where we measured neural responses across M experimental conditions, and we made these measurements across K different neural systems. So those are either K different animals or K different brain regions. And in each case, we kind of had this complicated high-dimensional measurement over N neural dimensions. And the hope is that we can boil this down into a simpler K by K distance matrix that summarizes all the similarities and differences. And then, again, what I'll claim to you is once we have that distance matrix, we're done, or at least very close to done, because there's a lot of things, a lot of off-the-shelf methods that are broadly available that directly work with data that is given to algorithms in this distance matrix format. So for example, I showed you mostly data visualization methods based on multidimensional scaling, or MDS. You can also run clustering algorithms directly on data that are represented in distance matrices like this. And you can also, it's not just unsupervised forms of analysis, you can do supervised regressions, such as K nearest neighbor regression. So I won't go into the details of all those downstream methods because there's too many to cover. But again, once we establish, or the argument that I'm making is that once we establish a good way of measuring distances between different neural systems, then we have a rigorous way to perform many different types of exciting downstream analyses. Okay? So in order to do that rigorously, what I'll now say is that it's ideal if you can define a notion of distance that satisfies some ground rules. And mathematicians call these distance functions, they give them a special name, they call them metrics. So a metric is just a notion of distance that satisfies a couple ground rules. The first is that the distance between 2 points, x and y, is equal to 0 if and only if x is somehow equivalent to y. So we'll talk about this notion of equivalence down the line a little bit. The second notion, or the second ground rule that we need to satisfy to create a metric space is have the function of distance be symmetric. So the distance from point x to point y is the same as the distance from point y to point x. Okay, so that's an intuitive condition. And then the third and final condition, which is usually the trickiest or most subtle to satisfy, is the so-called triangle inequality. And what this says is that the direct distance between any 2 points, x and z, is always less than the distance if I visit an intermediate point, y. So I took a direct flight here from New York to Lisbon, and that the distance that I travel by plane between New York and Lisbon is always going to be less than or equal to if I had a layover in London or any city in between. Okay. So these are the three rules that we want to satisfy to build a proper notion of distance. And the question that you should be immediately asking here is why? Just because mathematicians think this is a nice formalism for a distance, why should we as kind of practitioners in neuroscience care about this? My answer to that would be that many of the analyses I was alluding to earlier, such as clustering algorithms, regression algorithms, and data visualization, all of those, this slide it says only makes sense in metric spaces. I would say that it can be made much more rigorous in metric spaces. I'll soften that a little bit. So suppose, for example, that the triangle inequality is violated. What you can run into are situations like the following, which are bad news for clustering algorithms. If the triangle inequality gets violated, that means that you can find configurations of points where, for example, x is close to y and y is close to z, but at the same time, x and z are very far apart from each other. So this is the exact situation that the triangle inequality sort of precludes as possible. And in a situation like this, I would argue that a clustering algorithm faces a kind of paradoxical choice, because you would like to put x into the same cluster as y, and you would also like to put y into the same cluster as z, but at the same time, you would like to put x and z into different clusters. So if you were to satisfy the triangle inequality, then you would never run into a situation like this. And in fact, there's like a really nice transitive property of clustering that if x and y are in the same cluster and y and z are in the same cluster, then x and z have to be in the same cluster, and they also can't be too far apart from each other. Okay, so that's one intuition for why the triangle inequality is nice if you want to define an intuitive notion of a cluster of points. The triangle inequality is also really nice in terms of defining for each point who are my nearest neighbors. So for example, if I have many points, data points in a metric space, I can think about coloring those points by some dependent variable. So maybe each point in this metric space you can think of as a different animal, and I'm going to color the point by that animal's performance on a particular task. And now I make a neural recording from a new animal, and I'd like to ask the question, is that animal good or bad at this particular task? So one way to approach that would be to draw a ball or a circle around the new animal that I've just recorded, and then look at all other animals in my data set that are within this circular radius. Okay, and this is really easy to do within a metric space, but not so easy to do if you violate things like the triangle inequality. So once I've done this, now a simple estimator of how well this animal performs the task is to just take the average of all animals within this neighborhood, and that's called a nearest neighbor regression algorithm, which I have been alluding to before. Okay, so this is another form of analysis that's very computationally efficient and easy to implement in a metric space, and you need to be much more careful if you violate things like the triangle inequality. Okay, and then the final thing that's nice, and I'm not going to really leverage this in the examples throughout the tutorial today, but the other thing that's really nice about a metric space is that you can draw paths between points in a way that makes sense. So again, if I have points in a metric space, and I have 2 points x and y, you can again think of x and y as being two different animals in the data set. Under very mild conditions in most metric spaces, you can draw a path between these points where the sum of distances along this path is equal to the distance between x and y that we actually measure. And what that means is that I can actually do interpolation in this space, and I could ask, like, what is the most intermediate representation that's halfway between x and y? So again, the path length along this red curve is equal to the distance that that is defined on the metric space, and this means that interpolation and even extrapolation is easy to define in metric spaces, and it's really not so easy to define in non-metric spaces. You can extend this additionally to define things like averages and variances over the point cloud. And again, I won't talk about that in this tutorial, but these are, again, things that are really nice to have in metric spaces that you can't in non-metric spaces. Okay, so hopefully that answers the question, at least to some satisfaction, about why we're interested in defining these forms of distance. And so what I've told you so far is that we're going to define metric spaces over neural recordings, neural population responses. And when we do that, that's going to enable a pipeline like the one I just walked you through here, where we compute the pairwise distance between all neural systems we record, and then we hand off this distance matrix to a number of downstream analyses that can be used off the shelf. And I've showed you a couple example applications of this to visual coding and the head direction system. And the rest of the talk, we're just going to really focus on this idea of coming up with notions of distance that are interpretable and make sense to us as neuroscientists. And the challenge to that problem of coming up with a distance metric is that in neural recordings, neurons don't just come to us a priori matched or aligned across systems. So this problem would be completely hopeless if in each neural recording I only got one neuron from an individual system or animal. So if we were limited by 1990s recording technologies where you could only measure a few neurons simultaneously at once, this would not be tractable because the animal-to-animal variability would be completely swamped by sampling variability or measurement variability, because we just happen to record two different neurons with different tuning properties. So this idea of comparing across neural systems only becomes tractable, and this is really important, only becomes meaningful and tractable after you have access to many different neurons in each system, so that you might be able to say something rigorous about at the level of the neural population, are there statistical regularities here that are matched or mismatched across animals? And intuitively, one way to get at that would be to come up with some sort of matching or alignment of the neurons across one system or the other. So a number of the metrics that we talk about today are going to take this idea of finding a matching of the neurons, so that if I can find neurons in X that reappear in Y, and I can do that very reliably, then the distance between X and Y is going to be small. Okay, that's very high-level intuition. Okay, so yes, another question. I don't want to call these out. If Thanks.
Hey, I'm just wondering about the difference between finding a good metric for measuring distances and finding like latent dimensions where you can project these neurons into and then just use Euclidean distances. What's the difference between these two concepts?
Yeah, so the distances that we're going to end up defining are not going to be Euclidean. There's actually, this is getting too into the weeds, so maybe we can talk during the break, but the distances that we'll come up with end up having curvature to them. So it's not going to be so simple, you'll see.
Okay.
Yeah.
Thanks.
Good question. Okay. Thanks, Grant. Okay, so what am I saying? So the idea is that we're thinking about maybe aligning or matching neural dimensions across systems, and one way that hopefully a lot of people in this room are now familiar with thinking about this problem is in terms of the geometry of neural firing rate space. So for each neural system that we record n neural responses across m experimental conditions. This is a kind of canonical matrix valued data set that we're going to be thinking about today. Each row of this matrix I can plot in a high dimensional firing rate space. So again, hopefully people have seen this sort of thing before, where each neural dimension is the firing rate of a different neuron. And the first thing to maybe think about is a case where you have discrete experimental conditions. Like, for example, I show different visual images to the animal, and each image is going to correspond to a different point in the space. And I'm going to use colors to differentiate the points and use them as landmarks across different experimental tasks. Okay, So each experimental data set in this framework is a point cloud in a high dimensional space, and the points are labeled by condition labels. This also works not just with discrete categories, but if you have a continuous variable, like such as head direction that I was showing you before, you can discretize that continuous variable into a number of, in this case, head direction bins. And that will trace out some, again, low-dimensional, one-dimensional curve in neural firing rate space. And really, we're sampling points along that curve because we're going to incur some discretization. Okay, so this is a very flexible framework. I'd say that it applies both to continuous variables as well as completely different discrete categories. And we're going to think about the problem of aligning the neural dimensions across different tasks. So now if I record in animal X and animal Y, I get two of these point clouds in the high dimensional firing rate space. And the important thing is that the orientation of these point clouds is not a priori aligned because the neurons are not matched. So the neural dimensions do not come to us matched. And what that has the effect of doing geometrically is randomly orienting the point cloud. Okay, and at a high level, the idea of aligning neural representations is thinking about, can we fit a transformation, such as maybe a linear transformation, that would take the points in one space and align them to the other points. And now we see that actually the point cloud geometry is similar between these two systems. Okay, so that's a geometric interpretation of this idea of aligning neural representations. Okay, so the first idea for coming up with a measure of similarity might be to use linear regression to fit this alignment. So linear regression, the problem looks like this. I'm choosing a matrix R. I'm minimizing over R as a change of variables or a change of basis matrix that maps X onto Y, so that XR minus Y is as small as possible in terms of the sum of squared residuals. We have one question over there. Okay, so the first idea here is linear regression. And what I'll caution you now is that the linear regression for the types of analyses that I want to talk to you about today ends up not being sufficient because the underlying properties of the similarity score do not match up with that of a distance metric. And it's easy to see why linear prediction scores are not symmetric with each other. And I'm just going to go through this next slide and then take your question. I'll wait, actually. Yeah. So linear prediction scores are not symmetric and therefore violate like sort of core properties of a metric space. And it's easy to see why this is the case. If you think about a simple case like the head direction system, and imagine that I have two animals, one of which has no head direction tuning to particular heading directions. This is just a thought experiment. It's not something that we actually see experimentally. But if you were to imagine a mouse that had no responses or was sort of blind to 50% of head directions, I would be able to predict the responses in this mouse from a mouse that has tuning over all dimensions. But if I were to run the regression in the other direction and try to predict X from Y instead of predict Y from X, I would get a different R squared score because I fundamentally can't reconstruct all the different tuning curves in X from the subset of tuning curves that are observed in Y. Okay, so this is the quick intuition for why linear prediction scores are also are not symmetric and therefore aren't going to provide a metric space easily to us. This also works if you think about, this is just a simulation showing a similar effect in a simple toy neural network. So now I have X and Y are two 100-dimensional neural firing dimensions. And X is a very high-dimensional rich pattern of activity, and Y is a lower dimensional set of neural responses. So the way to think about this is if I do PCA on X, I'm going to need many, many PCs to explain all the variance in X, whereas for Y, the neural activity is constrained to a low-dimensional subspace. So in this example, when I predict y from x, I can do it almost perfectly. I get a r squared very close to 1. And if I run the regression in the other direction to try to predict x from y, again, I get a very different result. So again, I'm violating symmetry here. Yes, question.
Sorry, can you make any comment about the number of neurons that you have? Different animals, different brain regions will have.
How many neurons do you need to actually estimate this distance?
That it's different. That brain regions will have different number of neurons,
Yes,
and
yes.
different animals will. So then that will
so for
make
now,
you choose
yes.
which neurons to use.
Yes, great question. So yeah, how do you make comparisons if there's different numbers of neurons in each brain region? For linear regression, you actually could do that, right? Like you can make R. It doesn't need to be a square n by n matrix. I'm trying to simplify for now and just assume that we have the same number of neurons in each system. I'm going to very, very explicitly talk about this in the third part of the tutorial. There'll be like 10 or 20 minutes. Yeah, but great question. Okay, great. We're getting more questions, which I'm happy about. You can go first. We also have one over there.
Oh.
You go ahead.
Okay. So I just wanted to ask, is there a real difference between what you put as x versus y versus what you put as M conditions? It's actually the equivalent in terms of, like, just depending on what you're interested in.
Yeah, sorry. So what I should say is here, when we record from two systems, X and Y, I'm showing the same set of experimental conditions to those two systems. So the conditions are matched across recordings X and Y, but the neural dimensions are not matched. So those are the dimensions I'm trying to fit. Is that your question?
Not sure.
Not sure. Okay.
Ask again.
Okay. Yeah. Sure.
So you can imagine you can also put X and Y and Z and a lot of other animals as the M conditions here, and then you can change the condition into like the things that you're actually.
Oh, so like, so I'd have to think about how you do that. I think that would be a different form of analysis. What we're trying to do here is measure the distance between different animals. So for now, I'm just thinking about two animals, but if I have X, Y, Z, and many different animals, then what I'm going to end up doing is what's kind of summarized on this slide, where I'm going to compute all the pairwise distances between those k different animals and put it into this distance matrix. So the alignment task, I'm not thinking necessarily about predicting an animal, but that's an interesting idea, and maybe, we can chat more offline. Okay, one question over here.
Yeah, you were talking about the linear regression thing, and in the later slide you show the R, like we have to choose the R.
Yes.
But if
yes.
but if we go with the auto regression thing, then the residual error, that must be the fixed one. But why are you choosing this? I don't understand this.
Why am I? So this is the linear regression problem of finding the R that aligns X to Y as best as possible in terms of squared error.
Yeah, squared error. No, that error must be from the auto regression thing, or like what?
From the auto regression?
Like how are you deciding this R?
I'm choosing R to minimize the squared error between X times R and Y.
But how are you getting the error?
How am I weighting the error?
How are you getting the error?
Oh, I'm just asserting sum of squared errors as the loss function. Maybe I'm misunderstanding what you're getting at, and I can also take that question offline. Sorry. So yeah, this is just a simple idea of I'm trying to find an alignment between the neural dimensions and match these point clouds. So there's different loss functions you could use, but one loss function would be the sum of squared errors between the points in the line space. So how far away are the yellow points from each other, black points, blue points, etc. So that's just an example. It turns out to be the simplest loss function because you can have a closed form solution. We got more questions.
Yes. Why do you make the lack of symmetry the main obstacle? Because we could just take the even part of the distance and symmetrize the distance.
Yeah. So we'll find a way to symmetrize it in a second. Yeah.
Okay, thank you.
Yeah, symmetrizing it and then also finding something that satisfies the triangle inequality as well. So maybe I'll push on for a minute, but please keep asking questions. These are all really great. So there's a really simple way to rescue this approach of linear regression. So I told you linear regression is not sufficient because it doesn't give you a notion of distance that's symmetric. And it turns out that there's a simple constraint that you can add on top of a linear regression model that symmetrizes it and also gives you something that satisfies the triangle inequality. So the way to think about this is that it's the same objective function as before. I'm trying to minimize the squared error between the aligned representations, and I'm just going to add a constraint that the transformation R is what mathematicians call an orthogonal matrix. So that means R transpose R is equal to the identity. And geometrically, what this constraint means is that the rotation or the transformation from X onto Y is described by a rotation and reflection of the point cloud geometry. So I'm not allowed to stretch and compress dimensions when making the alignment. And intuitively, that was the problem with linear regression, that I could take a high dimensional representation and reduce it down to a lower dimension to predict a lower dimensional one, but I couldn't do the inverse of taking a low dimensional representation and expanding it to predict a higher dimensional one. Okay, so this problem, this constrained form of linear regression has a special name. It's called the Procrustes shape distance. And this is based on the idea that you're not changing the shape of the point clouds when doing the alignment. So when I take X and Y, I'm going to only fit a rotation and potentially a reflection of the points when making this alignment. And then I'm going to compute the, for example, root mean squared error of the points within the aligned space, and that gives me a proper notion of distance that is both symmetric and satisfies the triangle inequality. Question?
Yeah, so I have a question about like an alternative method to make the distance symmetric is to like you just add up the arrow of regress X onto Y and then the arrow of regress Y onto X. Is that worse than this method?
So that won't give you the triangle inequality. That will make it symmetric, though.
Okay.
Yeah, but intuitively, they're sort of doing similar things. Yeah, good question.
Is there an easy way to see whether it doesn't satisfy the triangular inequality?
I don't know of an easy one off the top of my head, but for the Procrustes distance, and I think this is what I have on this slide, you can prove that it's symmetric and satisfies the triangle inequality. And these are pretty simple proofs. They take half a page or less of math to show that they're symmetric and end up satisfying triangle inequality. Yeah.
Hi, thanks. So in the context of this linear prediction, when you say low dimensional, do you mean rank or is it more general than that?
That's what I was getting at, yeah, the approximate rank or participation ratio. If there's a big mismatch in the participation ratio or dimensionality, that's the situation where linear regression is going to be very asymmetric.
Okay, so it doesn't have to strictly be rank, it can be
It doesn't have to be the strict rank, yeah, no, it could be a soft notion of rank. Yeah, thanks, Sarah.
For Procrustes shape distance, we define dproc as RMSE in aligned space, right?
Yes. Yeah.
But what was the definition of distance in the standard linear regression case?
It could be root mean squared error or like 1 minus the R squared. You could think of it that way.
Okay, thank you.
Yeah, so it's the same, yeah. Literally the only difference that we're adding is this constraint of orthogonality. Okay, great, thanks.
Hi, can I ask why you don't just constrain R to be invertible?
Sorry, I couldn't hear that. Say again?
Why do you constrain R to be orthogonal and not just invertible?
If it's just invertible, then it's not symmetric. So that gives you the linear regression.
Okay,
yeah.
Thank you.
We will see how to build a metric that is invariant to invertible linear things in a second, though.
It's the rank of the data and not the transformation that matters.
The rank of the data?
Like, or the
So linear regression will be asymmetric when the rank of X and Y are very different,
right.
or X is low dimensional
okay.
and Y is high dimensional.
You,
yeah. Okay, great. I have a question over here. I'll take one more and then I'll move on.
Hi. I was just wondering, how do you deal with scaling? Because for me, if you take...
I'll talk about scaling next, yeah. Great question. Okay, so this is the Procrustes distance. You can think of it as just adding a constraint of orthogonality onto the simplest thing you could think of, linear prediction. And the other thing to note about Procrustes distance is that it's very easy to compute. It's built into the MATLAB statistics and ML toolbox. It's also built into SciPy, so it's not like this is some very complicated, difficult to compute distance metric. And the mathematically, what's happening under the hood here just involves like a simple singular value decomposition of the cross covariance matrix between x and y. So again, brushing the mathematical details of how you actually compute the optimal alignment under the rug, but it's not too complicated. Okay, if you're interested in Procrustes analysis and how it can be applied to neural data, I want to give a shameless plug for Grant's poster. Grant is one of our TAs here, and he's an experimentalist and has a really cool task where he trained mice to push a lever in a acoustically guided fashion. And then he has recordings in both auditory cortex and motor cortex during this task. And what he finds sort of remarkably is that there's a high degree of alignment in the representations between auditory cortex and motor cortex that emerges over the course of training in this task. So if you look at animals that are not skilled at the task, you don't actually see this alignment between auditory cortex and motor cortex, at least to the same extent. So that's just a very quick, shameless plug. I'm sorry for going over the really cool and important details of brushing aside a lot of the details that are on Grant's poster. But if you're interested in this, please go check out his poster at the conference. Okay, and then somebody asked me about scaling, and somebody else was getting at this idea that maybe we want not just rotations and reflections, but we want invertible linear transformation. So I'm going to get at both of those things right now. So the really cool thing about Procrustes distance is now that we have one way of defining a distance metric, it's easy to generalize it by allowing different pre-processing of your data before feeding it into the Procrustes algorithm. So generally, if phi is just some function, it could be any function like z-scoring or whatever, just some pre-processing function of our data, then it's sort of intuitively obvious that if I pre-process x and y, my two data sets, equivalently, and then compute the Procrustes distance on the pre-processed data sets, this also is a notion of distance that is symmetric and obeys the triangle inequality. And I'll walk through two different examples of pre-processing quickly. The first example is what people call the so-called classical Procrustes shape distance, and this deals with centering and uniformly rescaling. And then the second example that I'll walk through has a relationship to canonical correlations analysis, which is another method that you may have heard of for comparing representations. Okay, so for the first example, just think about simply centering and rescaling your data. You can write that mathematically as this function. You take x, you map it to c times x divided by the Euclidean norm of cx, where c is just a centering matrix. So c has this special form, but really you can think of this as just if you're in Python or MATLAB, you're taking x and you're subtracting off the mean of each column. So you're just recentering every column of x, and it happens to be the case that you can write this as a linear matrix multiply. Okay, so this is the pre-processing function that you would use, and geometrically you can think of this as if I have 2 point clouds, but they have similar shapes, but they are centered and they are rescaled at different points in firing rate space. I might want to, center them at the origin, or you almost certainly always want to center them at the origin because then you're fitting a rotation around the center of mass of the point clouds. So you almost always want to center at the origin, and if you rescale, you're making the distance measure invariant to like the overall size of the neural population. Okay, so this would be what people would call the classic Procrustes shape distance. So these centering and uniform rescaling operations are the sort of assumed classical pre-processing steps in what many people call Procrustes. Another example that you can choose that I think is interesting is the following one. So define x tilde to be this somewhat complicated expression, but it's basically just your centered representations, cx, multiplied by the inverse square root of this neuron by neuron covariance matrix. And what this has the property of doing is, again, centering the columns of x, so your tuning curves, centering your neural tuning curves, each at 0, and then rescaling them so that the covariance is equal to the identity. So you can think of this whitening transformation as just a multidimensional generalization of what z-scoring is. So after we apply this whitening, the mean of every single neuron is 0. The standard deviation of every neural tuning curve is 1, which is what z-scoring gives. But in addition, we're also reshaping the neural dimensions so that they're decorrelated from each other. Okay, so this is a well-known pre-processing step called a whitening transformation. And it turns out that if you whiten your data and then compute the Procrustes distance on your whitened responses, you get something that's sort of directly related to canonical correlations analysis, if you've heard of that before. So very briefly, canonical correlations analysis is a dimensionality reduction, or sometimes it's used for dimensionality reduction. It's an approach that gives you correlation coefficients across many orthogonal dimensions, essentially. And if you want to turn that into a distance metric, it turns out that the right thing to do is to take the average canonical correlation. So that's what I'm calling rho bar here. And then you can either take the arc cosine of that, or if you want to relate it to the Procrustes distance, you take 1 minus the average distance and take the square root. Okay. So again, mathematical details of this are something that you can look up in our papers and in the Jupyter notebooks. I think we link to those papers. But this has a nice, this pre-processing step has a nice feature that if I want the distance between my two neural representations to be 0, not just if there's a rotation and a reflection of the point clouds, but any invertible linear transformation, this is the right pre-processing step to build such a distance metric. So hopefully that answers, I think, what some of the questions we're getting at, which is what if I want a distance that is invariant or treats neural representations as equivalent if any linear transformation can relate them? Then this is the way to do that. Okay. And sorry, this is exactly what I just said, that the distance of this, the distance between these pre-processed representations is equal to 0 if and only if there's an invertible linear mapping, not necessarily a rotation and reflection, any linear mapping that maps x onto y. Okay, so geometrically, this is what this looks like. I'm showing 2 example point clouds in neural firing rate space. After whitening, you fit a rotational alignment and compute the root mean squared error in this aligned space. That gives you a distance metric that is closely related to the canonical correlations analysis. If you don't do that pre-processing step and just fit the rotational alignment on the original representations, you'll get a different notion of distance called the Procrustes distance. Okay, so that's just a geometric picture for the math on the previous slide. Okay, so I'm done with the first part of the talk. I want to summarize quickly, but hopefully I've whetted your appetite and made you excited about the possibility of using comparative analysis at the systems level in neuroscience. I think it's an exciting and emerging opportunity for the field. Hopefully I've also convinced you that having a proper notion of distance that is symmetric and satisfies the triangle inequality is a useful thing for downstream analyses such as clustering, visualizing data in low-dimensional spaces, and even performing supervised methods like nearest neighbor regression. It's not necessarily trivial to come up with these distance metrics, though, however. The simplest thing that you might try, just computing a linear regression, is not symmetric, and if you try to symmetrize it, it doesn't necessarily satisfy the triangle inequality. But there is a simple way to kind of rescue the idea of finding linear alignments between neural systems, and that's just to add a constraint on top of the linear regression, which is that the linear transformation has to be a rotation and a reflection, And this leads us to our first notion of distance called the Procrustes distance. And by the way, all the two example applications I showed at the beginning of this talk on the Allen Brain observatory data set and on the head direction, we're both using Procrustes distance. Okay, so we're running a little bit behind time, and we've been taking questions as we go, but if anybody has a question or two now, I'm happy to take them. Yeah. Yeah.
Could you give us an intuition about when to use Procrustes with versus without whitening? When would you want to use whitening and when not?
So I think the way that I'm going to come down on this by the end of the tutorial is that there's different metrics that you can come up with, and it's useful to try and compare them because it's probably not necessarily the case that like Procrustes is always better with or without the whitening. But you might try both and find, okay, actually the distance is really small when I whiten and it's much larger when I don't whiten. That suggests that there is differences in, for example, dimensionality that I need to account for. So I think it's not going to be the case that there's like a single best metric that you should always use, but you should maybe be aware of the different options and try different options. And sometimes the informative thing is actually, does the distance go up if I add this constraint or not, if that makes sense?
It does. Thanks.
Okay, great. I'm going to push on because I'll take one more.
Yeah, I'm trying to grasp intuitively if there is a secret ingredient or if we have anything we want with CCA. Like just by running CCA on X and Y, you look at this mean row, and you have the distance matrix, or is there something else in addition of that? Yeah, the average canonical correlation coefficient.
Yeah, okay, so yeah, here we're averaging over the canonical correlation coefficients, and you're saying that there might be actually individual structure in the individual canonical correlation dimensions.
No, it's just like the method you presented before with the orthogonal matrix.
Yep.
It's the same thing as running CCA between X and Y and taking the mean correlation coefficient.
Yes,
there is nothing more than that.
It's nothing more than that. Yeah.
Okay.
Yeah, exactly.
You just
This
presented
is
that
actually
it's
the way
equivalent mathematically. You just presented that the two are equivalent mathematically, but it's not a new method that.
It's not a new method.
Okay.
This is a nice reinterpretation of CCA. That,
okay.
Like you can think of CCA as whitening your data and then doing a Procrustes alignment.
Okay, thank you.
Exactly.
Thank you.
Yeah. Okay, one more, and then I'm going to move on.
Wait, so when I'm thinking about neural clusters and like comparing them, like if I want to do the distance between them, I'm thinking like do Procrustean distance because if you stretch it, then that's more like correlative instead of the actual distance between the two clusters. Is that accurate?
So when I'm talking about the clustering, I'm talking about clustering across different animals. Okay.
Yeah, I didn't mean clustering in that sense. I meant like a point cloud. So if you have two point clouds, why would you want to scale one of the point clouds to like sort of match the other one?
Yes. Okay. So going back to this example, is this a good idea to normalize the 2 point clouds to have the same size? And I would actually say, that is a choice, and it makes a lot of sense to me actually to not do that rescaling to the same size in Neuroscience, because we would actually want to know if one animal has high variance firing rates and the other has low variance firing rates. This rescaling step would actually try to hide that. So yes, the point is that you can do whatever pre-processing you want, and you should think carefully about it. And I think in many instances, it's probably a good idea not to rescale the size of the point clouds.
Oh, but will we cover cases where it would be a good idea?
Will we cover cases where
it's a good idea to rescale it?
No. I mean, we won't go into too many specific cases. Yeah, but yeah, this is just what people call the classic Procrustes distance. So I wanted to mention it. Okay, I'm going to move on to the next part of the talk, just to try to keep us a bit on schedule. I mean, we have 4 hours, and people are going to want a break in the middle. And we have more ideas to get to. So I'm going to continue by introducing another line of methods that some people in the room have probably heard about, including representational similarity analysis and centered kernel alignment, which is a different way of measuring similarity across systems. Okay, and to introduce or start this part of the talk, I want to take a step back and ask, why do we care about whether two neural systems have similar point cloud geometry in the first place? And this is explicitly what the Procrustes distance is trying to capture, is whether the shape of the manifold or the shape of the point cloud matches between two systems in a high-dimensional space. Okay? What I'll argue is that if two systems have similar point cloud geometry, then that implies that those two systems have similar, the linearly decodable information from those two systems is also similar. And that's at least one argument for why we as neuroscientists might care about geometric notions of similarity. So this part of the talk is mostly based on the work of Sarah Harvey and David Lipschitz, two of the TAs. And you can read about our thoughts, the details of this in the following paper. Okay, so here's the geometric intuition. I have two neural systems, X and Y. And in neuroscience, a common form of analysis that we do is to fit a decoder. So try to predict some output pattern from the neural responses in X and Y. So here I'm showing an example target pattern in green. And the first thing that I'll do is fit a linear decoder or fit a linear regression to predict this target pattern from the representations in X. And then I'll independently fit another linear decoder or regression to predict this green pattern from the representations in Y. And then the idea of this part of the talk is that we're going to look at comparing the predictions that these two linear decoders make and find that if two systems have similar point cloud geometries, then their predictions on this downstream task are also going to be similar to each other. Okay, so that's the intuition. All right, so formally, now I've introduced not just that we have two neural systems, X and Y, but we have an additional vector Z, which is serving as the decoder target. And if we restrict ourselves to linear decoders for now, the optimal linear decoder of predicting Z from either X or Y can be written in closed form. So here I've written down Wx as the optimal n-dimensional vector that will read out information from X to predict Z. And alpha and beta in this expression are just regularization terms that I'm not going to talk about too much. But you can set alpha and beta, for example, by cross-validation, or you can set them a priori by some other method. Okay, so that's the optimal linear readout from X. Alpha and beta are regularization parameters. And my prediction I'm going to write as Z hat X, so that's X times Wx. And then from the other system, Z hat of Y is the prediction from Y, which is, again, Y times Wy. All right, and now the really simple, dumb idea here is that if we want to define a metric over representations, one way to build a metric is to just look at the Euclidean distance between Z hat X and Z hat Y. Okay, this is very easy to prove that this is symmetric and satisfies the triangle inequality. Okay, so this is a new metric that I'm introducing to you now. And geometrically, you can think about it as follows. So I have two point clouds in a high-dimensional space. And now I've introduced this idea that I have a vector Z, which is my decoder target. And when I fit an optimal linear readout of X and Y, you can think of that as finding a dimension in X and an individual dimension in Y, such that when the points are projected and rescaled along this dimension, I'm reconstructing Z as optimally as possible in both cases. And intuitively, this distance that I'm defining, the distance between Z hat X and Z hat Y, is telling you how much the point cloud geometry agrees along these two dimensions. Okay, so that's a rough geometric intuition for this new metric that I'm introducing. Okay, I'm going to walk you through a few simple examples of this, because this is actually a way to generate many different kinds of metrics that might be targeted to the task that the animal is performing. So again, let's think about these simple kind of 3D Pringle chips as example representations. And I'll walk through 3 examples now. So in the first example, I'm trying to decode this kind of cosyne signal, Z1. And in this example, I'm able to do a very good job of linearly decoding the signal both from X and from Y. So in this plot, I'm showing the red and blue points are the predictions from Y and X respectively. And they're both closely matched to the signal I'm trying to decode, and they're closely on top of each other. So the distance between X and Y is going to be approximately 0 in this case, because their decoders agree along this decoder target dimension. Here's another example of a signal I could try to decode. And in this case, both X and Y do a terrible job of reconstructing this high frequency signal. So in this case, even though X and Y are both doing terribly, they're both doing terribly in the same way. So the distance between X and Y is again 0, because the distance between the red curve and the blue curve is very small. Okay, so that's a second example. Now, a third example of a decoding distance would be a task like this, and now something interesting happens. Both of the systems kind of are doing so-so at the task. They can decode it a little bit, but they're decoding and they're making errors in different ways. So now there are, you can see that the red and the blue curves disagree with each other, and therefore the distance in this third example would give a distance metric where X and Y are greater than 0 or further apart from each other. Okay, so to summarize all of this, this idea of defining a linear decoder and then measuring distance in the decoder prediction space is a very powerful and flexible way to generate different ways of comparing neural systems. And it's a way to really probe systems for information along particular tasks or dimensions. So for example, I can probe whether X and Y are able to make similar predictions on low frequency or high frequency signals in this simple example. Question back there. Okay. And one here first. Go ahead.
Does that mean it's better if I have like a the worse my information is decoded, the Z information is decoded in my population, the more information I get how similar the populations are? Because if they are perfectly reconstructing them, I just know that the information is in there, but it could be different mechanisms. It could be completely different populations that both have error 0, right? So I want to have them make errors, but in different amounts. Is that fair?
Well, I don't know what you want or don't want, but this, what I'm trying to show in this example is that the distance metric doesn't necessarily tell you if the networks are doing good or bad at the task. You have to separately assess that. So in the first example, they're both doing great at the decoding task, and the distance is 0. In the second example, they're both doing terribly, but the distance is still 0. And I think, again, the idea here from the first part of the talk is that I'm showing you all these distance metrics between just 2 example networks, but the ultimate goal is if I have 20 or so networks, I want to specify a metric like this and use it to compare many different networks at once. So it may be the case that for these 2 networks, neither of them does well, but if I had a third network, it does well at the second task on this example. And then the pairwise distances between networks are going to give me a sense of which networks are making similar predictions to the others on this task.
Okay, so I think what I'm getting at is essentially the 0 doesn't tell us much here, right? That's I think the question, because I could think of one rate-coded neuron that tries to encode a head direction versus a ring attractor. They both get perfect accuracy on the head direction, and then they have distance 0, but are completely different mechanisms.
I don't think 0 doesn't tell us nothing. I think, again, if you have, let's just say like 20 or 30 animals, and if the distance between some of them is 0 and the others are non-0, then that's interesting. So it's not. I mean, maybe I'm misunderstanding the example that you're thinking of, but what this is really defining is not just a distance between two networks, but it's a distance among many possible representations. And yes, if I have 30 different animals and they all have distance 0 to each other, that's not a particularly interesting metric space. I want to find metrics that are revealing similarities between some animals and differences with other animals. So a distance of 0 just means these two systems are similar on this task. And if across many different networks, I find some examples of individuals being similar and other examples of individuals being different, I would call that a win.
Okay, thanks.
Yeah.
Yeah. Do we have any guarantee that this is robust? I mean, like in number 3, is it possible there's a non-optimal readout vector that will actually match the errors between the two networks?
A non-optimal readout error. So you're worried about robustness to like outliers or robustness to something else?
No. So, right. I mean, the two errors here, the blue and the red ones, are different. You've optimized each of them independently to match the target, right?
Yes. Yep.
But it doesn't preclude the possibility that there is a linear readout from X that will exactly match that error of Y.
I think yes. So if the geometry between X and Y is similar, and we'll see this in a second, then there is no task that can separate them. If the point cloud, if the Procrustes distance is 0 between X and Y, let's say, there is no task that you can find that would erroneously make the distance large. One more question.
So far we are taking a noise-free picture of the system. We are taking average across many trials for each condition, right, to get the manifold.
Mm-hmm.
But when we think about decoding, often we think about inter-trial variability. And so you could have big distance with the same geometry if you increase the inter-trial variability for one manifold versus the other.
Sorry, the what variability? The
inter-trial variability. Inter-trial variability.
Yes, okay, fourth part of the talk. Although we might not get there if there's so many questions.
Okay.
Inter-trial variability is very interesting, and we are sweeping it under the rug for now. So for now, we're let's just say we're in a trial average regime, and we've averaged out all the trial-to-trial noise. Good question. Yeah, sorry. Okay, so let me give you a quick example application of this idea, which is going to be the subject of Sarah's Cosyne poster. So the motivation of this is taken from the field of neuro AI, where people are interested in making deep networks that are able to predict neural responses, either fMRI responses in human participants or maybe neural recordings in non-human experiments. And within this field, the Algonauts project and BrainScore are maybe two really popular ideas of this leaderboard style of building neural nets that are quote-unquote brain-like. So probably people have heard of these. An interesting problem has kind of arisen with these initiatives that's highlighted by this interesting paper by Colin Conwell and colleagues. This is coming out of Talia Konkel's group. And what they say in this paper is that if you look at the linear regression or predictivity score to predict neural responses from deep neural networks, specifically in this case they're looking at fMRI responses. Many different deep neural networks do equally well at this task. So there's two different measures of performance, but if you focus on this top one, which is just the raw linear predictivity score, you can see that even among like the worst neural network to the best neural network, there's barely 10% difference in performance. And their interpretation of this result in the paper is pretty interesting. What they say is many models achieve similarly high brain predictivity despite clear variation in their underlying representations, suggesting that standard methods used to link models to brains may be too flexible. And what they're getting at here is that when there's clear variation in the underlying representations is that there's two very different architectures being assessed in this study, which are vision transformers and convolutional neural networks, and they were kind of hoping to see a bigger difference between these two groups. Okay, so we can leverage this idea of decoding distance to take another look at this data set. So now we're using fMRI data from one of the human participants as our probe task. So the probe task is, given a neural firing rates in a high-dimensional space, can I linearly predict the fMRI voxel responses in a particular human subject? Okay, and now I can compute between all pairs of deep neural networks what is the distance between them using this decoding distance-based approach. So the distance between neural network I and neural network J is small to the extent that they make similar predictions about this human subject. So not necessarily that they're better or worse performing at the task, but whether they are making similar predictions on an image-by-image basis. Okay, so this is the pairwise distances. Remember from the first part of the talk, there's sort of a k by k distance matrix between all neural systems. That's what's being shown here. And you can do things like clustering and other downstream analyses. What I'm showing you here is just what I was showing you before of doing multidimensional scaling in PCA to visualize the data. So each point in this low dimensional space is a different deep network. And something that's cool about this experiment is that there was actually fMRI recordings from different human subjects. So we used one of the subjects as the target task, but then we can look at the other human subjects and put them into this metric space as well. So what you see in this space is that all the human subjects kind of cluster together, and then the deep neural networks are a little bit off to the side. They're not perfectly aligned with the human subjects. But importantly, the deep neural networks now kind of spread out within this higher dimensional metric space. And to some extent, you actually do see clustering of the deep neural networks by their architecture, whether they're vision transformers or convolutional networks, for example. So briefly, my argument would be that this metric space approach that I just walked you through gives us a richer and more detailed picture of what's going on when making comparisons across different deep neural network models, at least richer than just taking all the models and rank ordering them according to their predictivity scores on the human data. So I think this is, not necessarily replacing I'm not trying to say that we should do this instead of brain score or whatever, but this is a really nice complementary form of analysis to help us tease apart the finer scale similarities and differences across these different models. Okay. Okay, so far I've talked about linear decoding distances with respect to a particular distance or a particular target task that I have in mind. Again, the last example, it was predicting human subject fMRI responses. You might now ask the question, what if I don't have a good way of choosing the probe task, or I want to be agnostic to what the exact downstream task is of my neural representation? And one way to get at this would be to look at the average distance over many different random tasks that are defined on the data. So this is going to lead to what I'll call the average decoding distance. And the idea is to just sample your decoded signal Z from some distribution like like a white noise process. And what's very interesting is that if you then compute the average decoding distance, the number that you get out at the end of the day ends up being very closely related and I would say essentially equivalent to two well-known methods in the literature, which are representational similarity analysis and centered kernel alignment, RSA and CKA. So very quickly, RSA, or representational similarity analysis, for those who don't know, this is a very old idea, but it was popularized by this method by Nico Krieges-Korte and colleagues in 2008. It goes back much earlier to the 1970s. There's this paper by Shepard and Chipman that lays out this basic idea. And then many papers, kind of throughout the years have revisited this idea. But the short summary of what RSA does is it's going to look at point cloud geometry by looking at pairwise distances between points within the space. So if I have two neural systems X and Y, what RSA does is compute all the pairwise distances of points within X and all the pairwise distances between points in Y. So it calls these matrices dx and dy. It calls them representational dissimilarity matrices. And the idea is that These are number of conditions by number of conditions matrices. If these two matrices are exactly equal to each other, that means that the information is represented the same in these two systems, or their point cloud geometry is exactly the same. Okay, so to summarize how similar these pairwise distance matrices are, one classic way that this is done in RSA is to just take the correlation, the raw uncentered correlation between dx and dy. So this is the mathematical expression for that. You can just think of this as take dx and dy and turn them into vectors and compute their normalized inner product. It's sort of the uncentered Pearson correlation between dx and dy. Okay, so that's a very popular, there's variants of RSA, but this is a very popular way of looking at RSA or implementing RSA. And then there's another very popular approach that people may have heard of called centered kernel alignment, or CKA. So this is very popular in the machine learning community. It was popularized by this paper by Simon Kornblith et al. In 2019. And it was inspired by some earlier work within the machine learning community, not going back to the 1970s, but still this idea had kind of been floating around a little bit for a while. Okay, but what's interesting about this is that CKA is almost exclusively used by people in the machine learning community and people who study deep learning and publish in NeurIPS, ICML, and ICLR. Very few of them will use RSA. They'll use centered kernel alignment instead. So there's kind of two different fields and a limited amount of cross-citation between those fields. Here's the idea behind centered kernel alignment. Instead of taking all the pairwise distances between points in this high-dimensional space, I'm going to look at the angle between points. Or really what I'm going to look at is the dot product between all pairs of points in this high-dimensional space. Okay, so I'm going to call those kernel matrices and call them kx and ky. If I'm just using a linear kernel, this is going to be the same as a covariance matrix. You can also think of kx and ky as covariance matrices. Okay, so these are, again, number of conditions by number of conditions matrices. And the idea is if kx and ky are identical, the point cloud geometry is identical. Okay, so in centered kernel alignment, as implied by its name, you don't just use the raw correlation between kx and ky, you include a centering operation. So remember from the first part of the talk, we have this centering matrix C, which all it does is mean center either the rows or the columns. In this case, you're multiplying on both sides of the matrix, so you actually center both the means and the columns of the kernel matrix. Okay, but just think of this similarity score that CKA gives you as a kind of centered correlation coefficient. And that's one of the things that distinguishes it from RSA. So to summarize, in RSA, it's very popular in the psychology and neuroscience literature. You're looking at the pairwise distances between points, and you're using the uncentered correlation score between these representational distance matrices. In centered kernel alignment, this is much more popular in machine learning. It's using inner products between object pairs rather than pairwise distances, and it uses a centered correlation score. So there's a centering operation that's also introduced. Okay, so intuitively, these are kind of similar methods because they're looking at pairwise distance or similarity scores between points within each neural representation space, but they give different numbers. So people have kind of treated these as being different methods in the literature. But it turns out that if you incorporate the centering operation into the RSA score, you get these two things to actually agree to each other. So the centered RSA score is equal to the centered kernel alignment CKA score if you choose a distance and kernel function that are compatible with each other. So the distance and the kernel have to satisfy this really simple relationship. But what this basically means is that you can hand me your favorite distance function and say, okay, I computed RSA with this distance function. Can you find me the kernel that I would use to create the same distance function or the same similarity score with centered kernel alignment? So this is, you can hand me a distance function, I can give you a kernel, or vice versa, and I can get these two methods to agree to each other exactly. The only thing that you need to change is you need to incorporate a centering step into RSA, and if people are interested in why you might want to include a centering step into RSA. I talk about it a little bit in this paper, and I also give a quick proof of this fact. Okay, so as an interim summary, I talked at first about linear decoding distances as a way of comparing high-dimensional representations with respect to a single decoder target. And then I told you about these two other methods that kind of came out of left field, right? So RSA is an old method where you're looking at pairwise distances between points, and CKA is a kind of newer method that came out of the deep learning community that uses pairwise dot products instead of pairwise distance scores. And what I just got done telling you is that the RSA and CKA score can largely be unified once you incorporate a centering step into those two. And what I will tell you about next is how you can relate both of these to the decoding distance. And I know I've sped up because I'm trying to keep us on time, but we have a question over here. Yeah, go ahead.
Can you give us a little bit idea about how does the centering idea work in the RSA? Because the distance matrix, in my opinion, is not dependent on the center, right?
Yeah, okay, so the reason why centering you might argue centering is a good idea on RSA is that the raw correlation, so distance matrices are non-negative. So if I take the raw correlation, uncentered correlation between dx and dy, it can never actually be 0. So it's scaled between, it's not actually scaled between 0 and 1. If I do the centering step on the distance matrices first, then I get a similarity score that actually goes between 0 and 1. That's the intuition I can give.
But to my opinion, the centering operation, you just move the axis, the coordinates from some original points to another point.
Yeah, sorry. So you're not centering the point clouds. What you're actually, and I'm sorry that I went through this fast. You center the matrices themselves. In this case, the kernel matrices. And in the distance, you're centering the dx and dy matrices. So dx and dy are the pairwise distances between points. Those are all non-negative. And the centering operation centers them, makes them positive and negative so that you can take their inner product and actually get a 0 out.
Okay.
Does that make sense? Okay.
Thank you.
Yeah.
So you're forming these matrices based on M conditioned by N neurons data. Yep. But if you have like time varying data, where does the time dimension goes?
Yeah, I'm going to talk about dynamics a bit in the fourth part of the talk. A simple way to incorporate time is to just treat the time bin as a condition. So if I have like an event-triggered response, I could take M time bins after that event-triggered response. I think that's a perfectly fine way to incorporate time. And there are some more exotic choices that I'll talk about in the fourth part of the talk. Okay, great. Quick question over here, maybe?
It's just really quick. So this, the kernel measure and the distance measure, can we still apply everything if the space is not Euclidean? If
if you use a nonlinear kernel?
If I need the geodesic distance to measure in the interdistance or the angle between, for example.
Yeah. So there are nonlinear extensions to the CKA where you use a nonlinear kernel. For geodesic distances are not actually super easy to relate to positive definite kernel functions, so there's some subtle differences there, but maybe happy to chat more about those offline. Yeah, good question. Okay, so I've told you about, so far, 4 different metrics: the decoding distances, RSA, CKA, and in the first part of the talk, the Procrustes shape distance. I just got done telling you that you can really think of CKA and RSA as the same thing. They're very closely related to each other. And now what I want to tell you about is an interesting connection between the decoding distance that I told you before and the CKA and RSA scores. So let's go back to the linear decoding distance framework that I talked about before. So here, remember, we have point clouds of neural recordings from animal X and animal Y predicting target pattern Z. And if I'm fitting a linear decoder, I can write down in closed form what is the optimal prediction that you get from X. I'll call that Z hat of X. And the optimal linear prediction you get from a decoder fit to y, I'll call z hat of y. And the interesting thing here is that you can write down a closed form solution for the optimal linear decoder, and it's actually a linear function of z. So I'm just going to take this long matrix at the beginning and call it kx and ky. So you can think of the optimal prediction z hat of x and z hat of y as just a matrix multiply with z given by this matrix kx and ky. Okay, so this is what I just said, that the linear decoding distance is actually just a distance between kx minus ky multiplied along a dimension z. Okay, and kx and ky, again, just to put them back up there, are these kernel matrices. You can write them down as XX transpose and YY transpose. So these are pairwise dot products, just like in the CKA setup that I was showing before, where X tilde and Y tilde are regularized whitened response matrices. So you can think of this as a pre-processing step where I take my data points X and Y and I apply some whitening transformation, or maybe I don't want to apply a full whitening transformation because that often has numerical stability issues, kind of divide by zero errors. So I can add these regularization parameters alpha and beta to my whitening operation. And then I can compute kx and ky and measure their distance along a particular dimension z. And now you might believe me that if I average over all possible z's, what I get then is just the Euclidean distance essentially between kx and ky. And that gives me another distance metric, which I'll use capital D to denote and call this the average decoding distance. This is the average agreement between decoders over essentially all possible tasks, where those tasks are drawn from a white noise process. Okay, so that's a little bit of math and extension of the linear decoding distance, but what's very interesting is that this new distance metric that I just sketched out for you is closely related to centered kernel alignment and therefore also closely related to representational similarity analysis. Okay, so the geometric picture, let me just give a geometric picture to go along with that math. I have point clouds in two neural firing rate spaces. And now instead of having a single decoding target, I'm sampling many random decoding targets, Z1, Z2, Z3, etc. And for each one of those, I'm fitting an optimal linear decoder independently to X and Y, and then measuring the agreement between the geometry of X and Y along those readout dimensions in blue. So as I sample more and more target patterns Z randomly from a white noise process, I'm getting snapshots essentially of what the point cloud geometry looks like along different dimensions in the neural firing rate space. So it's at least intuitive that as I sample more and more random tasks or more and more dimensions, I'm checking whether the shape of X and Y agrees along all those dimensions. Okay, so that's the intuition for what's happening now in this average decoding distance where I'm not using a single task, but I'm looking over many tasks. And intuitively, if the average decoding distance between X and Y is very close to 0, then that must mean that X and Y have similar shapes in their point cloud geometry within the high dimensional space. And the punchline is actually that the average decoding score is essentially interchangeable. I won't say equal to, because CKA and RSA are usually similarity scores rather than distance scores. But there's a very simple relationship. It's just basically 1 minus the centered kernel alignment score between this average decoding distance and CKA. So this is the relationship that you get at the end of the day at the bottom. And what that basically means is that I like to think of CKA and RSA scores as being interpreted as what is the average decoding similarity of my two neural representations over a random set of tasks. Okay, so one way of summarizing this part of the talk is that it's a new interpretation of what CKA and what RSA are actually measuring. Okay, and then the big picture summary before we're going to have a break is that I've told you about many different measures of similarity so far, And in fact, there's even more. There's, for example, this review paper, which comes out of the deep learning community that catalogs over 30 different ways that people 30 different quantitative measures people use to quantify similarity in high dimensional neural representations. And the good news that I've been trying to tell you today is that many of those measures that exist within the literature are essentially equivalent to each other. They differ by, for example, a centering operation in the case of RSA and CKA. So we've unified those. And then also, many of these measures, I think, can feel a bit abstract at first. They're just measuring similarity in the geometry of high-dimensional point clouds. But you can actually directly relate the geometry of these high-dimensional point clouds to what is the linearly decodable information from these neural representations. So we can relate it to decoding analyses, which is something that neuroscientists are very used to and comfortable with. So to summarize, I started out by making that point or making that connection between geometry and decodable information. I showed you that you can build targeted probe tasks to define metrics over neural representations with respect to particular task variables. So this is a very flexible way of designing particular distance metrics. I showed you an application of that to analyzing fMRI data and comparing it to comparing deep neural network representations within that framework. And then in the second part of the talk, I related this idea of decoding similarity to two very well-known methods that people have probably heard of, namely RSA and CKA. And you can think of RSA and CKA as measuring the average distance between decoders over many, many tasks. And then finally, there's one loose end we have, which is, I told you at the beginning about this Procrustes distance metric and how is that related to the decodable information in RSA CKA. The really short answer that I'll give here before giving us a break is that it turns out that the Procrustes distance is a more stringent notion of similarity, so that if you have a small Procrustes distance between two neural representations, that means that the RSA or CKA similarity score is very high. But the converse is not necessarily true. If you have a high RSA CKA score, you don't necessarily have a small Procrustes distance. So the Procrustes distance is a more stringent notion or stringent condition for judging similarity. And this was pointed out in one paper by Nathan Kloos and colleagues, where they tried to look at what are the different metrics that you can get in terms of Procrustes similarity and RSA CKA similarity, and they find this region of envelope. We also looked at this in our paper. We took a more mathematical approach and actually proved some upper and lower bounds on how big the Procrustes distance can be if the RSA CKA distance is small or big. So you can check out these two papers for more details. And yeah, let's all take a break for coffee. I can hang around and answer some questions if people have any. And let's come back in like 20, 30 minutes. That's just linear regression. And then when I build up this new notion of distance, I'm distance here. Okay. I'm going to get started again. Running 10 minutes late, which is not too bad, but hopefully everybody had enough time at the to get a little bit of coffee, maybe play with the Jupyter notebooks. I'm going to give people a minute or two to get settled in their seats and then get started. On this website. They have a lot of animations in them, but okay. So while people are coming back inside and getting settled down, I'll also just flash up and remind you that we have some Jupyter notebooks and Colab notebooks available at this website that'll remain up after the conference. So please take a look at them if you're interested in using these methods. It'll kind of walk you through step by step how to compute these measures on data. And also give you a little bit, I think, in some instances, a little bit of mathematical detail behind how you actually compute them. Okay. Okay, great. So I'm going to continue now with the second half of the tutorial. And the way I want to start off this half is by mentioning that all the methods that I told you about in the first half of the talk were built kind of explicitly to be invariant to rotations of the neural firing rate space. And this is most explicit, I think, in the Procrustes shape distance, where you're comparing point cloud geometry between neural systems by fitting a rotation and a reflection of the neural axes. So obviously the distance is equal to 0 here if there is some rotation that aligns the point clouds. This is also true for the methods that I just told you about in the second part of the talk, namely RSA, CKA, and all of the decoding-based different distances that I mentioned. In all of those cases, if you just rotate the point cloud geometry, that doesn't change the output of a decoder because I can just rotate the decoder weights. And it also doesn't change the RSA, CKA summary statistics. When I rotate those point clouds, it doesn't change the pairwise dot products or the pairwise distances between these points. Okay, so all of these are invariant to rotation. And I want to start out by asking the question, is that actually the right assumption, at least for certain scientific questions? And to get intuition for why rotations might be too flexible of a class of transformation, you can think about tuning curves such as these ones on the screen now. So these are sparse unimodal tuning curves. You see things that look like this in visual cortex, orientation selectivity, for example. You also see things often that look like this in head direction systems where there's a unimodal bump of tuning to head direction. Okay, so if I now think about what this looks like, what these tuning curves look like in the neural firing rate space, they're going to trace out a one-dimensional trajectory, as I mentioned before, and in this simple example, it looks like this. Now, if I apply a random rotation to this manifold of neural activity and project this back down onto the 3 coordinate dimensions, I'm going to get a rotated basis, a rotated set of tuning curves that'll look like this on the right. And the point is that if you look at these two systems at the level of their tuning curves, they actually look like they're organizing informations quite differently. But from the standpoint of the Procrustes distance and all of the decoding-based distances that we talked about, the distance between these two neural representations is going to be, they're going to be quantified as being 0 under all those measures. Okay? And if you look at how neural systems are organized in many cases, you see that they often are organized along particular neural dimensions in a structured way. So I already alluded to the fact that in early visual cortex, you get orientation selective neurons. This is obviously a very well-known and cited result. This is also true in higher order sensory areas, like, for example, when you find face selective neurons in certain areas of the brain. And this isn't just true in biological systems, it's also true in deep artificial neural networks. So this is from an example paper showing an example unit inside a hidden layer of a deep artificial network and showing all the images to which that unit had a positive response. And you can see that this unit more or less responds to all images with legs or even horse legs, and a couple other images in red in the lower right that also have leg-like features. So this paper called these object detector units because they're very specific and fire selectively when a particular kind of object is present in the image. So this is something from that completely from the deep learning community, completely independently of the neuroscience literature. This is also true. Another way to see this in deep artificial neural networks is to take the first layer weights of one of these networks. Like, for example, AlexNet, which is a very well-known early deep convolutional neural network for image recognition. If you take the first layer weights and just visualize them, you get things that look like oriented edge detectors or other detectors for differences in color and center surround structure. So these are the first layer filter weights of AlexNet, and if you apply a random rotation within that neural space and then re-express those filter weights, you get something that looks like this, and it would be hard to argue that these two systems are actually equivalent to each other. Okay, so all of this is kind of pointing to the idea that, at least for studying certain kinds of questions and for certain, under certain sets of assumptions, rotations are maybe too flexible of a way to compare neural systems. Okay, so let's reevaluate the Procrustes distance in light of this observation. So the Procrustes distance, remember, is a constrained form of linear regression where I'm minimizing some alignment matrix R to align X to Y, subject to a constraint that R must be a rotation, must implement a rotation and a reflection, i.e. be an orthogonal distance matrix. So now I'll introduce a variant of the Procrustes distance, or an extension of the Procrustes distance, sometimes called the permutation Procrustes distance, and also maybe called the one-to-one matching distance. So the only difference that I've introduced here is this new constraint on the lower right portion of the slide, which is that the alignment matrix P has to be non-negative. I can't have any negative entries. It still has to be an orthogonal matrix, so P transpose P has to be the identity, but in addition, it needs to be non-negative. And this extra constraint is enough to constrain P to be what's called a permutation matrix, and that's why I'm using P instead of R. So R was for rotation, and P is for permutation. This permutation Procrustes distance is still a tractable and computable measure, and it's easy to show that it's symmetric and it obeys the triangle inequality. And this is what the alignment matrices look like under these two situations. So on the left, I'm showing you an orthogonal matrix, just a random orthogonal matrix. You can see that it has positive and negative elements, and this is the sort of thing that the Procrustes distance or the Procrustes alignment procedure would return a matrix like this to you when trying to align neural representations. On the right side of the screen, I'm showing you a permutation matrix, which again has the same constraint that P transpose P is equal to the identity with a non-negativity constraint. And that non-negativity constraint is enough to completely constrain this matrix so that it only has zeros and ones. And there's only a single one in each row and column of this matrix. That's another way of describing what a permutation matrix is. So this is called a permutation matrix because when I multiply a vector by this matrix, it just reorders or permutes the order of the elements of that vector. Okay, so let's give an explicit example of what permutation versus orthogonal alignments look like in a simple neural system. So here I have three neural systems, X, Y, and Z, and I'm looking at tuning along a single dimension, and the vertical dimension are different neurons. Okay? So if I try to find the optimal alignment of x to y and z where I'm only allowed a permutation or I get to reorder the neurons, it's maybe intuitive that I can't align x onto y. So you can see the blue line and the black lines are not matched in the middle. But I can find a permutation of x to align it very closely to z in this example. So z has very similar tuning curves to x, and therefore I can find just a way to reorder the neurons in x to match it to z. If I were to do the Procrustes distance instead and fit an alignment, it's possible for me to align x either to y or z. So again, this is a looser notion of distance or a more permissive alignment class of transformations. And if you think about the underlying metric spaces that this induces, the permutation Procrustes distance would therefore say that neural system X and neural system Z have a distance close to 0. They're very close together. And system Y is very different from X and Z. Alternatively, if you use the classic Procrustes distance, which fits a rotation, this would have the effect of saying that system X, system Y, and system Z are all very close together. So these are two different notions of a metric space that give you different insights or different clusterings of what is similar versus different in these neural representations. Okay? Both of these are proper notions of distance, though, and they respect the triangle inequality. Okay, so how might we leverage this idea to ask a specific scientific question? So I want to do that over the next couple of slides by asking the question, are neural axes in firing rate space preserved across systems? So let's say, just to draw you another picture, I have two systems X and Y, and I'm going to consider not just X and Y, but a randomly rotated copy of the X matrix. So here I'm doing a full rotation in the n-dimensional firing rate space. And now I want to consider what happens if I fit the optimal permutation alignment between these systems. So I'm going to use permutation-based alignments. Permutation-based alignments in this geometric picture just mean that I get to relabel the coordinate axes, but I don't get to rotate and reflect the point cloud. So first I'll show you y aligned to x. This is just a schematic, but if I align y to x, maybe I can find y and x have a high degree of alignment according to the just relabeling the neurons. But no matter what I do, I can never really align y to x that's been randomly rotated. So xr is the randomly rotated copy of x. And because I've randomly rotated the point cloud, I've destroyed any of the special structure or alignment that may have pre-existed in X along the neural dimensions. Okay? So what I just walked you through, the logic of what I just walked you through is that if the neural axes are somehow special and they're preserved across systems X and Y, or another way of saying that is if the neural tuning curves are special and preserved across X and Y, then by randomly rotating X, I'm going to decrease the similarity or equivalently increase the distance under a permutation-based alignment, because that random rotation is intuitively destroying all of the structure of the tuning curves and mixing them together. So you can think of XR as a kind of shuffle control for this analysis. And what's interesting is that if you apply this to real neural data sets and also to deep neural networks, you often find that such random rotations do decrease the similarity score under permutation-based alignments. So that's what I'm showing you here in these two figures. These are an example comparing two brains to each other, to human brains under fMRI or to monkey brains with neural recordings. This is also true if you do the comparison across two deep neural networks that are trained on a task. And if you are interested in more of this result, I'll give a shout out to our paper that's in press at Nature Human Behavior. This is work that was led by Meenakshi Khosla and in collaboration with Josh McDermott and Nancy Canwisher. Okay, so I'm just showing you a very quick result from that paper, but the upshot of that result is that there is often sparse and axis-aligned tuning in the data sets that we looked at least. So throughout this tutorial, when I show you point cloud geometries in this high dimensional space, I've been kind of scattering the points evenly and naively across this space. But really, when a more realistic picture, at least for the many sensory systems that we looked at, is that the point clouds are kind of structured in a way that is reliable with respect to the neural coordinate axes. And you get neurons that are kind of sparsely active to different conditions, but detecting different features, for example. And this isn't the point of the tutorial to really go down the rabbit hole of what this might mean or what is the bigger picture for neural coding, but you can imagine that this sort of a representation where information is preferentially aligned along the neural axes might have certain advantages, and people have thought about this for a long time in terms of energy efficiency or the ability to efficiently read out information through a sparse decoder. Okay, so I won't go into that, but these are interesting consequences of this form of analysis. Okay, so that was a very quick introduction for how you can incorporate a more stringent constraint onto the Procrustes distance that doesn't allow for rotations, but actually tries to find a one-to-one matching or permutation of the neurons to align systems. And now the question that probably many of you have is, how do I apply that to a system where I have different numbers of neurons? And that's what I'm going to talk about in the rest of this talk, or the rest of this part of the third part of the tutorial. Okay, so the problem is, how do we handle neural systems with an unequal size? And this is a problem both for the permutation Procrustes distance. I can only fit a permutation matrix if the permutation matrix is square. And it's also a problem with the original Procrustes distance, at least how I've presented it to you. So I can only fit rotational alignment if it's an n by n, n-dimensional neural matrix. So even though we like to conceptually think about comparing representations between systems with equal numbers of neurons because it simplifies things, in reality, this is almost never the case, and we need to really think about this problem seriously. Okay, so this is what we're going to focus on. Okay, and just to make it clear why this is a problem, let's think about the problem of matching tuning curves across these two example neural systems. And one of them has 8 neurons, one of them has 3. So there's a big mismatch in the size of the recorded population. Okay, so I can find tuning curves that perfectly match from the black population onto the blue population, the tuning exactly on a one-to-one basis. So the question is the distance between these two systems 0, or is it something else? And I'd argue that it's not immediately obvious how you should think about the distance between these two systems, just given the information I've told you. In particular, if I'm just greedily matching units one-to-one, and I'm allowed to leave some of the units unmatched in the larger system, which is what I've done, then it's very easy for me to find matches between a big system and a small system. So systems that I just happen to record lots of neurons in are going to be easier, or going to have a distance that's close to 0 just by nature of having more information available within them for the matching process. So again, we want to come back to this idea of, can we build up notions of distance that are symmetric and which satisfy the triangle inequality, and how to deal with this fact that in neuroscience experiments with electrophysiological recordings, for example, you just have a sampling bias problem. In some animals, when you record, you'll just record fewer neurons. Okay, so there's a very, I think, elegant solution to these problems, both for the permutation-based alignments as well as for the Procrustes distance. And one way of getting around these problems is to reframe all of these alignment problems as distances between tuning curve distributions rather than alignments of neural firing rate spaces. So that's what I'm going to walk you through right now. Okay, so the geometric picture I've been showing you thus far, I've shown you this again and again and again. You have X and Y are two matrices, and you can plot the rows of those matrices as points in an n-dimensional firing rate space. But because the neurons are different in the two systems, the point cloud is randomly oriented with respect to each other. So there's no alignment between these two n-dimensional firing rate spaces. That's the problem we've been focused on. Equivalently, rather than plotting the rows of x and y, I can think of plotting the columns of x and y. And now something interesting happens. Each column of x and y is a point in an m-dimensional space. So each axis here is now a different condition, and each point in this space corresponds to a neuron. So I only have a single space. I don't need to learn an alignment between two spaces. However, the points within this aligned space are no longer labeled because, again, the neurons are not one-to-one matched. So each neuron corresponds to a point in this space. So if this is a 3D space, each point would tell me how much each neuron responded to each of the three conditions, and that would give me a point within that space. And I'm coloring all the neurons from one system, Y, in red, and all the neurons from the other system, X, in blue, but I can't do any coloring or labeling beyond that because the neuron labels are arbitrary. Okay, so you can refer to these two views of the data as a few different ways. I'll use firing rate space to refer to the geometric picture on the left, and I'll use tuning curve space to refer to the picture on the right. And I'll often use this kind of fancy word that mathematicians use called duality to say that these are two equivalent representations of the same underlying mathematical object. So each one of these systems is equivalent, or each one of these ways of looking at the data is equivalent to the other. Okay, so let's think about this problem of matching individual neurons in tuning curve space. So I'll show you a simple example where each system has 4 neurons. So we're still working in the regime where the two systems are matched in terms of the number of neurons. And I'll plot this in 2 dimensions. So let's just say we record how these neurons respond across 2 experimental conditions. So now I have 4 points in a 2-dimensional space. That's the tuning curve space that I just walked you through. Okay, and finding the optimal one-to-one matching or the permutation matching that minimizes the squared error between these representations basically means that I'm finding a way to connect each blue point to a red point, and I want to minimize the overall path length of these black lines that are connecting the points. Okay, so this would be the optimal matching of the points that I'm showing you here. There are other ways to match the points that would be suboptimal and have a larger root mean squared error when summed across the whole population. Okay? Okay, so that's the permutation matching problem in tuning curve space. And what's really nice is that this problem I'm going to show you now generalizes to cases where you have different numbers of samples and different numbers of blue points and red points. This is a problem known as discrete optimal transport that's very well studied within the mathematical community. So it's called optimal transport for the following reason. One motivation for this is to think about you have a set of resources that are spread across warehouses in some environment. So let each one of these red points, think of it as a warehouse, and think of each blue point as a factory that needs material from that warehouse. If I set up the problem so that each warehouse holds exactly 1 unit of resource and each factory requires 1 unit of resource, you can ask, what is the optimal way for me to transport my material from my warehouses into my factory? So find me the cheapest way to transport resources into the factories, and this will give you, in this case, the optimal permutation between the warehouses and factories, the red points and the blue points. So there's no need to split resources and redistribute it across systems. There's just match each warehouse to its closest factory, more or less, and then proceed with that transport plan. Okay, so that's an interpretation of this discrete optimal transport problem. And the nice thing is that this works with an unequal number of warehouses and factories, which in our setting of neuroscience is going to mean that this works with an unequal number of neurons in each system. So here's just a simple example where I have 4 warehouses and 2 factories. And I'm going to set up the problem as follows. I'm going to say nx is the number of warehouses, just to be general. And I'm going to evenly distribute my resource units across all warehouses. So each warehouse holds 1 over nx resource units, and the total amount of resource units sums to 1. And I'm going to let ny be the number of factories. That's the number of blue points. And assume that the factories evenly require the resource. So each factory requires 1 over ny resource units. Okay? So the problem, again, is to find the cheapest transport plan. And in order to do this, you need to allow yourself to either split or recombine resources across the warehouses and factories. So in this case, the optimal thing to do is to transport data along these transport resources along these black lines, which will satisfy the constraints that we're working with. The resource will get to the factories that it's needed, and the overall transport cost is minimized. Okay, so that's just an intuition. This overall problem of optimal transport, it's called, is extremely well studied. It was first kind of developed in the Soviet Union, in the World War II era Soviet Union, where they were trying to move troops around in an efficient way, or move resources around in an efficient way. And now there's very tractable algorithms for solving this sort of problem. Okay, so that is a little bit of a side or abstract background on discrete optimal transport. The important thing for us as neuroscientists is that this works for finding matchings of neurons across systems. So this works in m-dimensional tuning curve space. We treat the warehouses as neurons from one network, and we treat the factory as neurons from a second network. And what this yields is a soft matching or a soft permutation of neurons. So before, when I was trying to match neurons across this system, systems with unequal sizes of neurons, I was greedily matching neurons one at a time and allowing some of the neurons to be mismatched. The optimal transport soft matching distance is a really nice generalization of this that ends up being symmetric and satisfies the triangle inequality when I compute this across many different neural systems. And it doesn't allow you to drop units that are dissimilar from one network to the other. So every unit has to be matched, at least to some extent, to neurons in the other network. And you get a soft or weighted permutation or matching of the units as shown here on the right. Okay, so this is a really nice way to reframe the problem of aligning representations by permuting the coordinate axes to aligning or measuring distance within tuning curve space. So I showed you this to begin with. We have firing rate spaces with M labeled point conditions, and then we need to fit an alignment, either a rotational alignment through Procrustes, or implicitly fit some sort of alignment or coupling between these spaces using a decoding task. That's what we talked about in the first 2 parts of the talk. Now I've introduced this idea that you can instead reframe the problem as measuring distances between unlabeled points in tuning curve space. So now there's a single space where you can plot both x and y, and each point in this space corresponds to a neuron on the right side of the screen, whereas on the left side of the screen, the points are labeled and correspond to conditions. All right, and I call this tuning curve space. You have n unlabeled points or neurons, and what I just kind of got done showing you is an example of how you can use a notion of distance called an optimal transport distance to compute the distance between these point clouds in this tuning curve space. And really, you can think about this as what I'm actually trying to do is compute the distance between some underlying probability distribution over the neural tuning curve. So as I sample more and more neurons from neural system X, I'm getting more and more samples from this distribution in blue. And as I record more and more neurons in system Y, I'm getting more samples from this distribution Y. And to the extent that the tuning curve distributions agree with each other, I'm saying that the information is being represented similarly between those 2 systems. So that's another way of thinking about this reframing of the problem. Is that on? Yeah.
now it's on. How does this measure is not penalizing the fact that if I compare one system with, I don't know, 10 neurons and the other one with 3, and two systems with only 3 neurons? Because the 3 to 10 seems that have much more distances to measure, while if you had another system with 3 neurons in this example, the overall sum of distances might be smaller.
It's appropriately normalized by this normalization here. So if you divide by 1 over nx and 1 over ny, so there are different numbers of neurons, but they each carry equal or like a weighted amount of importance that ends up being consistent.
Okay, perfect. Thank you.
And I guess I should say that here, when I compute the soft matching on systems of different sizes, I'll get a soft permutation. But if I were to compare 3 versus 3, like you were saying, I will actually get a honest to goodness hard permutation matrix out of that. So this is a true generalization of the permutation Procrustes distance. Yes, over there.
Thanks. I was wondering, given your emphasis on the tuning curve, does it mean that we can use this alignment only if we are dealing with sensory systems?
No, sorry. So when I say tuning curve, I'm using it pretty generally. Like the tuning curve could be across time bins, for example. I mean, people in motor cortex also used to talk about tuning curves a lot back in the day.
You need to know the function of those neurons, otherwise it's not applicable anymore.
Yeah, sorry. I don't need to know the function per se, but I need to sample the function over a discrete set of m points. So yeah.
I was wondering if instead to map each individual dot with each other, like we do with the optimal transport, could we just look at the statistics of the distribution by trying to compare the mean and the different moments of each distribution?
Yes. Yeah. So there's these two ways of thinking about neural representations. This is going to be a direct answer to your question. And what I just got done telling you was that this permutation Procrustes distance, where I fit an alignment where I just get to relabel or permute the coordinate axes of the firing rate spaces, this is equivalent to an optimal transport problem where you find the optimal permutation between points in tuning curve space. And this is useful because that dual representation also generalizes to cases where you have different numbers of neurons. But these are two more or less equivalent notions of distance. If I evaluate them, they're going to give me the exact same distance score. So that's two ways of thinking about the same distance score. And now you're asking, are there other notions other metrics that I can interpret in this way? And that's exactly right. So it turns out that if I now think about the classic Procrustes distance, where I'm not just fitting a permutation of the units, but I'm fitting a rotation and a reflection of the point cloud, the classic Procrustes distance you can show is equivalent to the following notion of distance, which is called the Burris distance or Burris-Wasserstein distance, which is the optimal transport distance between two Gaussian distributions. So the way to think about that is I have two point clouds in this tuning curve space, but I don't want to individually match the points one-to-one to each other. Instead, I'm going to just look at the mean and covariance of those distributions and then compute the optimal transport distance on these Gaussian distributions. It's really the mean-centered Gaussian distribution, so that you're only looking at the distance between their covariance matrices. So exactly what you're saying, I just compute the covariance matrices in the tuning curve space and have a notion of distance on the covariance matrices.
But can we generalize it to any order of moments, like not first moment and second moment? We could do that for
So any distance between probability distributions is probably a reasonable way to put a distance metric on neural representations. I think that's right. Yeah. Over here.
So just sticking with this probability interpretation, is there a way in which you can kind of connect these ideas to a notion of comparing two equally sized data sets, but where one of them is just missing some data, and then some of the neurons, for example, and then you just kind of marginalize out over
Good question,
those tuning
yeah.
curves? You see what I mean?
Yeah, I don't have any slides on missing data, but absolutely you could. I think you don't necessarily need to leverage the duality to get around that problem, but there's definitely ways of making or estimating these distance functions under partial observations or missing data? Yeah, great question.
Cool, thanks.
Okay.
Where's the question coming from? Back
Thank
here.
you.
Yeah.
I want to know if this method provides a symmetric matrix. Is symmetric?
Which distance? The soft matching is symmetric?
Yeah, that's.
Yeah. So the distance is now just your transport distance, like in the example here. So it's the length, the weighted length of these black curves, black lines, and that will be symmetric in the sense that the distance from x to y is the same as the distance from y to x. So one way to think about that is if I swapped the location of the warehouses and factories perfectly, the transport plan would just proceed in reverse. So the transport distance would be the same. Does that answer your question?
Okay, I know, thank you.
Yeah. One more.
So before you were talking about sparse and axis-aligned representations, like in sensory systems. And so when you're imagining comparing two different systems, they may have some shared and some distinct component. Can this disambiguate those? Like, will it tell you that some axes can be aligned, some modes of one population? And
great question. Again, I'm not going to talk about it for the purposes of this talk, but so within this tutorial, I'm really focused on this problem of defining a distance measure between neural population responses. But some of those distance measures, such as this soft matching distance and the Procrustes distance in the first part, those give you a kind of alignment transformation, or in this case, a soft permutation that has information embedded in it that tells you which units are matched to the other units. So for the purposes of this tutorial, or at least what I was telling you in the first part of the tutorial, where you're building up this distance matrix across all systems, and then using that for multidimensional scaling and downstream analysis, we often just throw away this alignment information. But it's absolutely true that there's interesting structure in there and that you could do some sort of secondary analysis on to supplement what these metrics are telling you. Great question. Can I push on for 5 or 10 more minutes? I just want to make sure that we don't miss Chris Ola's talk later. Which we might. And I want to go to his talk, so I'm getting, I don't know about the rest of you, but I'm getting sick of listening to me. Okay, so the permutation Procrustes distance is equivalent and has this dual relationship with optimal transport in the tuning curve space. It turns out that the classic Procrustes distance, I'm not going to prove this to you, but it's not too hard to prove, that the classic Procrustes distance is equal to a distance between covariance matrices that you fit in the tuning curve space. So that's another relationship which is really nice, and this also means that you can compute the Procrustes distance on systems with unequal number of neurons. And then finally, in the second part of the talk, I kind of snuck in this duality relationship for you already. So I'd argue that the average decoding distance that I told you about, so remember here, we are looking at similarity in the predictions of linear decoders, and each linear decoder is defined by a dimension z, and if I average over random dimension z, I get a notion of distance that was equal to centered kernel alignment, or RSA. That was the kind of punchline right before the break. And if you look mathematically at what I wrote down in the second part of the talk, the RSA CKA distances that we were looking at are just distances between the covariance matrices in this tuning curve space. And the average decoding distance is a distance on the in firing rate space where the projections are taken along these dimensions W that are fit by linear decoding. So W is this n-dimensional or number of neurons dimension. The length of vector W is the number of neurons. So that is happening in the firing rate space, and it is dual to a distance between covariances within this tuning curve space, which corresponds to CKA RSA. Okay, so to summarize this part of the talk, I told you that permutation Procrustes distance is equivalent to this optimal transport distance or soft matching distance. You can see this paper for more details on that. There's also, I mentioned and didn't prove to you, an equivalence between the classic Procrustes distance and a distance between covariances in the dual space. That's shown in this paper. And then finally, in the second part of the talk, I also showed a duality relationship between the average decoding distance and RSA CKA type distances. So this final paper is the one to read for more details there. Very quickly, I want to have one slide on why I think duality is useful to know about. The first is that, as I told you before, it solves this conundrum of if I have different numbers of neurons in my two systems, am I really doing the right thing to compare systems? And in the dual space, you just have different numbers of samples from these probability distributions over tuning curves. And then it somehow, like, I think very elegantly puts that problem to bed by saying that it doesn't matter if I have different numbers of samples from these two distributions. What I'm actually trying to estimate is the distance between the underlying distributions. The other reason this is useful is that it shows that alignment methods, such as the Procrustes distance, which operate in neural firing rate space, the n-dimensional neural firing rate space, are often really not that different from similarity comparisons such as CKA and RSA, which happen in the m-dimensional tuning curve space. And then a third and maybe more subtle point is that there's actually complementary strengths to the thinking about things in firing rate space versus tuning curve space. Sometimes it's quicker to implement certain algorithms in one versus the other, or it's easier to mathematically prove things in one space versus the other. Okay, so this is summarizing the third part of the talk, and then I'll have some water and take some questions. What I started out by telling you is that most measures of representational similarity are rotation invariant. This is true for the Procrustes distance and RSA, CKA, and decoding distances. But we know that in many neural systems, representations are not really rotation invariant. There's information that is organized along particular neural dimensions. So I showed you that, for example, by scrambling the filter weights in AlexNet. And I also, again, am giving a shout out to our paper that's in press at Nature Human Behavior that tries to show this systematically. And one way that we show that systematically is by developing metrics that compare representations that are explicitly sensitive to rotation. So it's easy to, for example, extend the Procrustes distance by incorporating a non-negativity constraint that means that the alignment between neural dimensions is completely sparse and one-to-one. So this is sometimes called the permutation Procrustes distance. Okay? And then in the second half of the part 3, I walked you through this idea of duality and how all of these metrics can be generalized and computed in cases where you have different numbers of neurons in each system. So for example, for the permutation-based distances I was talking about, you can read this paper that I wrote with Meenakshi on soft matching distance. And the deeper point leveraged this idea that we can, this deeper duality between labeled points in n-dimensional neural firing rate spaces versus unlabeled points in a unified tuning curve space. These are two geometric views of the same problem that we've been talking about this entire time. And great, I'll take some questions now if there's any in the audience. We have one here. Sure.
So when we just talk about the alignment is in a stable time point, like we don't care about the manifold dynamics.
Yeah, people want to hear about dynamics. So I will talk about dynamics in the fourth part. A simple way to deal with the dynamics is to just treat each condition as a different time bin. So this is a very flexible framework that's just deals with comparing labeled points across a set of recordings, and if those labeled points are just labeled by the time bin condition, then that's one way to deal with that with dynamics.
Thank you so much.
We have one up here. Maybe you can ask and I can restate the question.
Yeah, it's
sorry, go ahead. Yeah, so the question is about, in addition to rotation, can we think about stretching and shrinking? And yeah, so in the part one of this talk, I mentioned stretching and shrinking. Like the way that I would typically handle that is through a pre-processing step of whitening, if you want to be invariant to stretching and shrinking. So yeah.
My question is about how important is the fact that the minimization problem to find the matrix P is non-convex?
It's convex.
Ah, it's convex.
Oh, sorry. There's a convex relaxation. I should be precise. There, and I'm brushing all this under the rug. There's a convex relaxation, and the solution to that convex relaxation gives you the solution to the problem that you care about.
Okay.
Yeah.
Thank you.
So it's a very, very elegant mathematical theory. Yeah. One over here.
Is it my turn? I have a related question to the other two questions in the regimes where the conditions are time bins.
Yep.
When we don't have a stimuli or a structured task.
Yep.
The problem with those data sets are usually the number of neurons and number of time bins are very, very large.
Yep.
And I can see how to generalize these methods to those regimes, but the problem is you run into an undersampling problem because you are working with, say, 10,000 dimensional spaces. I was curious if these methods would work under those circumstances, or any extension of these ideas for those kind of
So I'm going to talk
data?
about sampling error in the fourth part of the talk. So yeah, hopefully I'll answer that, and if not, maybe you could ask again, because I'm going to talk about both of those things in the fourth part. Is there any other questions? Okay. I'm gonna. Okay, one last one, and then.
Sorry.
And then we have 15 minutes.
Yeah. So can we intuitively understand the soft matching as we so for the sample with fewer neurons, can we understand them like I'm mixing some neurons from the larger sample size?
Yeah, that's right. So I mean, basically with the soft matching, in order to make it satisfy the triangle inequality and be a self-consistent distance metric about across 3 or more representations, what you need to do is you need to enforce that every neuron gets at least partially matched to something, to a neuron in the other set. So I don't know if I go back far enough. The problem with this greedy matching approach, no, I went too far. Yeah, so the problem with this greedy matching approach, which is shown on this slide, would mean that the black neural representation is going to have a distance that's very close to 0 to everything else if it always has more neurons. Because if it has extraneous neurons that are not matched to any other network, then it's not being penalized for those extra dimensions. That's maybe an intuition. So the soft matching requires, or it penalizes a network for having extra extraneous tuning curves that are not matched by enforcing a matching. Okay, great. So I'll move on to the fourth and final part of the talk, and then hopefully have time to take more questions at the end. So in this final part of the talk, I'm going to give a brief overview of a lot of things that I consider pretty cutting edge methods within this space of comparing neural representation. So hopefully if I pull this off right, I'm going to raise fewer questions than I'm able to answer, and I'll inspire people in the audience to come back next year and have great Cosyne posters that solve some of these open problems in the field. Okay, so one open problem that that we have some results on, but I think is still a challenge, is how do we actually apply these methods in practice? And this has come up several times, that neurons in reality are noisy. So if I show the same stimulus, or if I have the animal perform the same behavior from trial to trial, neural spike counts or firing rate responses are going to be stochastic and fluctuate from trial to trial. Also, when we make a recording in a particular brain area, we only get to record a small fraction of the neurons within that brain area, and we're kind of going in blind. We're randomly sampling a subset of neurons within the brain area. And then finally, we only get to record neural activity over a relatively limited amount of time, and we can only probe a limited set of experimental conditions, so we can't exhaustively sample conditions either. So all of these are going to intuitively lead to some sort of estimation error in the comparison metrics that I'm actually trying to estimate between neural systems. Okay, so I'll start with the noisiness from trial to trial and how we think about that a little bit. So thus far, I presented this sort of naive picture that you have a discrete set of conditions, and each condition is mapped deterministically to a point in high-dimensional neural firing rate space. If I were to mathematically formalize this, you would think of the neural system as a simple input-output map, mapping stimulus inputs or behavioral outputs to a unique response in the neural firing rate space. So if UI is the condition on, or the input to the system in condition I, then the neural response would be given by xi, and I get to sample these n-dimensional vectors xi over 1 to m experimental conditions. And this is what we've been talking about so far. So relaxing this and making it more realistic, we can think about what happens when neural responses are noisy and probabilistic. So the appropriate generalization here is that the neural system is not a deterministic input-output map, but it's really a conditional probability distribution. So conditioned on some stimulus or motor behavior U, you would draw a neural response from this underlying probability distribution. And I would sample this across conditions UI, where I goes from 1 to m, and then I would sample perhaps multiple trials per condition. So I denote the trials by k going from 1 to capital K. Okay? And commonly, what's commonly done currently within the field is to look at trial average responses and average away the noise as much as possible. So the geometric picture to have in mind would be I have some set of experimental conditions. In each condition, I sample multiple trials, so I get multiple points of the same color. And then I average away all that noise as best as possible. And now I have 2-point clouds that I can treat as deterministic point clouds and just apply all of the methods that I was telling you about, Procrustes, RSA, CKA, et cetera. Okay, so this is most commonly what is actually done. And I think, as several people in the audience have been pointing out, this ignores some of the rich structure within these data sets. So I'll come back to that later, but that's at least a very simple and easy way to deal with the problem of noisy responses across trials. So there's also these other two problems that I mentioned, which is that we only get to observe a subsample of the neurons and a discrete number of conditions, which might not be all different conditions that the neural system is exposed to in everyday life. So let me draw you a couple geometric pictures of this problem. So we can think of X and Y as in their firing rate space. We've seen this picture again and again. And really, when we run our experiment, we're sampling experimental conditions as discrete conditions from an underlying distribution or a space that is continuous and has many rich experimental conditions. So if my inputs to the system can be continuously tuned, or the behavior of the animal is continuous in a naturalistic environment, the possible neural firing rates that you would actually see over all naturalistic behaviors, at least in an idealization, people usually think about this as a continuous tuning or a continuous manifold in neural firing rate space. So What we actually want to do when we're comparing neural representations is comparing similarity between these full manifolds, but all we get to measure in an experiment with a finite amount of time is we get to measure discrete points over this manifold. So you can think of these as landmark points that are capturing something about the geometry of the manifolds, but only imperfectly. Okay? So that's the first problem of subsampling conditions. And then the second problem, where we only get to see a subsample of neurons, you can think of this as killing off certain dimensions. So here I'm showing that by removing the vertical dimension from this plot. So when we record a subsample of neurons within a brain area, you can think of this as projecting the landmark points onto a lower dimensional space. And then this final thing on the right-hand side of the screen is actually what you get to measure, and it's what you get to compare in terms of the Procrustes distance, RSA, CKA distance, etc. Okay? Just because I got done talking about duality, let's quickly go through the equivalent dual space view of this in terms of tuning curves. So you have two systems, X and Y, which we can think of as distributions over tuning curve space. When we only get to measure a subset of possible experimental conditions, you can think of that as now projecting out or killing off certain dimensions of this tuning curve space. And then when we sample neurons, you can think of sampling neurons as drawing samples from these probability distributions. So we'd like to estimate, what we'd actually like to estimate is the distance between the probability distributions on the left side of the slide. What we actually have access to are samples from a low dimensional projection of these probability distributions, and that's going to lead to some sort of error and distortion. Okay, so this is all just walking you through some geometric intuition for how subsampling conditions and subsampling neurons can impact these distance measures. But the question is, how many neurons and conditions do we need so that we're doing a good enough job of sampling or covering the space of conditions and neurons all the way on the left side of the screen? Okay? So intuitively, yeah, if we sample enough neurons and enough experimental conditions, the things that we actually compute actually do approximate the things that we would like to compute in an ideal world. Okay, and there's several papers that deal with this problem of sampling error, and most of the work that I'm aware of, at least, has tackled this problem individually, of assuming that you have a fixed number of experimental conditions and then just look at how the error changes as you sample more and more neurons. And then other work has focused on the other problem of, let's say you have a fixed number of neurons and look at the error as you change the number of experimental conditions. So you can read all of these works if you're interested in that. I'm going to give a quick plug for upcoming in-progress work that my student Cosyne is working on, which shows that, which tries to do something more holistic and looks at how does error grow or how does error decrease as you increase both n and m simultaneously, and trace out the full two-dimensional error landscape that looks something like this. And again, this is an unpublished result, but one of the things that we're thinking that we're seeing is that in many of these distance metrics, the scaling is pretty favorable, even in complex high-dimensional data sets. The expected error incurred by subsampling neurons and conditions is only proportional to 1 over m and 1 over n, plus 1 over n. So you get an error landscape that looks like this on the right, and as you increase the number of neurons and conditions, you get a better and better estimate. Okay, and this is actually pretty favorable scaling compared to other problems that neuroscientists are interested in, like inferring the curvature of neural manifolds, for example, is actually harder than this problem. Okay, so stay tuned. I think there's more work to be done there. For those of you who are very, you're practically minded, you want to go home and you want to apply this tomorrow, and you don't want to wait a year or two for Cosyne and me to figure this out more comprehensively. How do you quickly check that you have enough neurons and conditions in practice? What I would say very quickly is that I think neuroscientists have gotten pretty good at developing cross-validation procedures and shuffle controls, and you can do this for a lot of the metrics that I mentioned. So for example, For the Procrustes alignment, you can run cross-validation specifically on the alignment learned by Procrustes and tests whether it generalized. Another thing that I mentioned in the very first part of the talk was this idea of splitting your neural population randomly from each animal into two subpopulations and then measuring the distance of an animal to itself as a kind of baseline null control. And what you see is that the distance from an animal to a different animal is usually actually larger than the distance of an animal to itself when judged by this split population control. And then there's other ways of doing shuffle controls and bootstrapping over your data sets. There's a nice paper on this from Nico Krieges-Corte's group. This is work led by Heiko Schutt, who now has his own group. So you can also check out this paper, which deals specifically with the case of representational similarity analysis, but introduces and deals with bootstrapping procedures to get error bars on these estimates. Okay. So hopefully that gives you a little bit of a satisfactory answers to points 2 and 3, which is that as we record more and more neurons and we record more and more experimental conditions, we believe that these distance metrics will converge to the quote-unquote right thing. But I kind of brushed over the first problem a little bit, that neurons are noisy from trial to trial. So in the final part of this tutorial, I want to return to this problem of noise from trial to trial and also ask a lot of the questions that have been coming up about neural dynamics. Okay. Any questions? Good. Okay, so what do we lose by looking only at trial averaged responses and not looking at trial by trial fluctuations? The answer I would say is that potentially we lose a lot by doing this, even though it's a very common thing to do with this sort of analysis. So in particular, it's easy to imagine coming up with two neural systems that have identical trial average geometry, but have very different noise profiles, and therefore, if you think about them from the standpoint of whether their functional utility or downstream readouts, they would perform very differently. So for example, here's X and Y. These are two systems that have, I haven't done the rotational misalignment here to make it easy for you to see that their trial average geometry is exactly the same. But if the trial noise is very different across these two, it would be hard for you to argue that X and Y are equal or equivalent to each other. So in particular, if I think about training a decoder to distinguish different task labels or condition labels from each other, network X is going to have poor discriminability or decodability between the three colors on the left, and Y is going to have poor discriminability or decodability among the three conditions on the right side of this plot. So all of this is completely missed by trial averaging responses. And moreover, there's just a rich history in the history of computational neuroscience on modeling the geometry of noise in neural systems and relating this to decodable information. This is a really nice review paper from 2006 by Averbeck, Latham, and Pouget, Nature Reviews Neuroscience. What they're showing in this figure from that paper is just you have two stimulus conditions and you have noisy probabilistic responses. So we're thinking of these as multivariate Gaussians. On the left side of the screen, there's very little correlation between the two neural dimensions. So you get circular contours of the probability distribution. So without noise correlations there's a good separability between the conditions. On the right side of the screen, you've just introduced noise correlations between neuron 1 and neuron 2, which leads to this elongated ellipsoidal contours. And even though the total variance or noisiness is equal in these two cases, the decodability goes down when you introduce noise correlations into the system. And there's many really influential and classic works on this idea of looking at noisy co-fluctuations between neurons and how they limit the information content of systems. Okay, so for all of that, let's think about how we can extend some of the ideas we talked about today to handle noisy trial-by-trial variability and some of this structure. So This work that I'll describe now was co-led by Lyndon Duong and Jingyang Zhao. And this is the paper that you can read to read more about it. And the basic idea is that we want to fit the Procrustes distance, but not just to the means, the trial averages of these distributions, but looking at the mean and covariance of each of these distributions. So we're going to fit a rotation and a reflection to align the two point clouds. But now when we do the alignment, we're not minimizing just the Euclidean distance between the means. We need to generalize the Procrustes distance to measure the distance between distributions in the aligned space. So there's two different ways that you can think about doing this. You can use a Wasserstein distance, which is an optimal transport distance between distributions. And then there's also another notion of distance called the energy distance, which is just a different way of coupling the two distributions and measuring a distance. So the details of that don't matter too much, but the point is we want to find a rotation and a reflection that aligns these colored distribution sets as best as possible and then measures their distance in a reasonable way. And both of these distance measures on probability distribution are true generalizations of the Procrustes distance, so that as the variance of these point clouds shrinks and each point becomes a delta function at the trial average, this would reduce to the Procrustes distance. So these are true bona fide generalizations of the Procrustes distance. Okay? So that's, we call this the stochastic Procrustes shape distance. And in the final part of the talk, I'll show that you can use this simple idea to get at some of the other questions that have been coming up related to looking at neural dynamics. And in particular, I'll think about this really thorny problem of disentangling input-driven versus recurrently driven dynamics in recurrent neural circuits. So this is work that was led by David Lipschitz and Amin Najakbash. And there's two different papers. One is a workshop paper, one's a ICLR paper that go into the specifics of these methods. Okay, so we were really inspired and motivated by this interesting example that was published by Anirudh Galgali, Manish Sahani, and Valerio Monte. So they show this simple example of, and they motivate it with a decision-making task. So the idea is I'm recording in, for example, prefrontal cortex, and the animal is deciding between making choice 1 or choice 2. So it's a binary choice task. And there's a couple different canonical models of how choice can be supported by these circuits in prefrontal cortex. So in particular, one classic model that a lot of people attribute to Xiaozheng Wang is the idea of a bistable system where you have a saddle point in the middle and then two attractive fixed points that pull the neural dynamics towards choice 1 or towards choice 2. So that's a bistable saddle point system on the left. Another canonical model would be a line attractor model where I'm slowly integrating evidence towards one end of the firing rate space versus the other, and I'm slowly accumulating evidence towards choice 1 versus choice 2. So it's not bistable, it's you have a whole manifold of marginally stable fixed points along a line. And then they look at a third example, which is a point attractor. So the idea here is that everything just collapses to the middle, but it's entirely driven by the inputs of the circuit. So across all three of these examples, what they do is they adversarially tune the inputs to the recurrent neural network to drive it to have the same exact trial average behavior. So the idea is by looking at the trial averages here, you have no information to distinguish any of these three flow fields from each other. They all have the same trial average behavior, and that's because the inputs have been, the input drive to the systems has been adversarially tuned. Okay, so if you model the neural system with the following equation, so x is the firing rate vector, and it's updated according to x of t plus 1 is equal to xt plus some nonlinear recurrent function f of x plus input drive u plus noise. That's the model. Then the result is that the trial average activity cannot disentangle recurrent dynamics from inputs because the trial average activity, which I'm denoting m here, is equal to this combination of the expectation of f of x across trials and the input drive u. And you can adversarially tune u to overcome the effect of the recurrent dynamics. So f of x is encoding the recurrent flow field, and it's mixed in with the input drive u here in terms of the trial average activity. However, the interesting result is that the noise covariance in this simple model does not explicitly, at least, depend on the input drive to the system. So if you look at the covariance of the trial-to-trial covariance across neurons at time t, it obeys this update equation, where you'll see u of t never even enters into this update equation. So in principle, at least, you can use the trial-to-trial noise to help distinguish the different recurrent flow field dynamics. And just to really emphasize this point, the fact that the trial average activity cannot disentangle recurrent dynamics from input, and the noise covariance can, means it's really impossible to think about disentangling input-driven dynamics unless you look carefully at the trial-to-trial fluctuations or you apply some perturbation to the system from trial to trial. So I think it's really important to emphasize that if you're interested in recurrent flow fields, you need to be thinking about noise or perturbations to actually resolve what those flow fields look like. Okay, so here's the quick result showing you simulations across these three systems. So at the top, I'm showing you trajectories of individual trials, and at the bottom, I'm showing you the trial average as points, and the covariances, the trial average covariance as ellipses within this low-dimensional space. And you can see, as before, the trial average is exactly the same, but the covariances actually are different and can be used to distinguish the simulations that were published in the original Galgali et al. Paper. So when you apply the trial average Procrustes distance, it's unable to distinguish any of these three systems from each other. But if you add a stochastic dimension to the Procrustes distance, as I described, then the stochastic variant of the Procrustes distance actually can tell these systems apart from each other. Okay, but then David and Amin took this analysis even further and said, well, maybe the noise covariance at any individual moment in time still might not be enough. So in the follow-up paper, what they did was they constructed examples where the trial-to-trial mean and the trial-to-trial covariance at a particular point in time were indistinguishable between two systems. So this is an example of that. So I have neural population A and neural population B. And if you look at their aligned trial averages, the trial averages are more or less exactly the same. And this is also true if you look at each moment in time, what is the trial-by-trial noise within the system. So the covariance ellipses here in blue and red are also very closely overlapping. However, if you look at the individual trajectories on the left-hand side of the screen, you can see that there's a very obvious and big difference between population A and population B. So the idea was, well, maybe we need to compare these systems by using all of the information across the full trajectory. So the idea is instead to compare systems across their full trajectory. Okay, so now the way to think about the full trajectory now is to think of each neural circuit as encoding a distribution over noisy trajectories. So xk is going to be a number of neurons by number of time bins matrix on trial k, and that's sampled from some distribution p of x, which maybe depends on the experimental condition that the animal is in on trial k. And I get to measure these noisy n-dimensional distributions across multiple trials. And xk is a random matrix, as I said. And then the goal now is to come up with a distance between these neural systems, which are encoded as distributions over noisy trajectories. And I'll just give you a brief flavor of how we do that. The first step is to learn the mean and covariance of px and py by some method. And then the second step is to compute a form of the stochastic Procrustes distance, and there's a couple different variants of this on those means and covariances. So the important thing to mention here is that the mean and covariance that I'm referring to is actually so the mean is an n times t dimensional matrix, And the covariance is an NT by NT covariance matrix in this kind of expanded space of neurons and time bins. And if you use all of that information across the full trajectory, then you can actually distinguish these cases, even in more adversarial cases where the trial average as well as the individual covariances at each time point are equivalent to each other. So here I'm just showing you that the Procrustes distance is unable to distinguish these three flow fields. The stochastic Procrustes distance, where you just look at the covariance at each moment in time, is also not able to distinguish these systems by construction. But if you use the full trajectory information, as I just kind of briefly sketched for you on the previous slide, you are able to distinguish and find that the saddle point line attractor and point attractor have different statistics in the statistics over noisy trajectories. Okay, very briefly, I have one slide on other methods for comparing neural dynamics. Another popular method that has been gaining traction within the field is this dynamical similarity analysis. This is a paper by Mitchell-Ostro et al. From 2003. Just very briefly, the idea there is to use Koopman operators or analysis to characterize nonlinear dynamical systems and approximate those operators using delay coordinates. And then they use a Procrustes-like transformation to compute the distance. But at least in the original formulation of the problem, there's no explicit noise model within the recurrent dynamics. So that's a difference between the approach that we took. Another paper that I quite like is this paper by Sagodi and Park, which they call dynamical archetype analysis. And here the idea isn't necessarily to come up with a proper metric over neural representations, but instead come up with a library of canonical dynamical computations, so like line attractors, point attractors, saddle points, and use these dynamical archetypes and stitch them together to stitch together these building blocks, essentially, to build a recurrent flow field and use that to compare neural systems. One downside of this is that it requires that you have access to the ground truth vector field, which would be available to you in an RNN model, but it's hard to really get at this in biological data, as far as I can tell, at least. Okay, so summarizing this part of the talk, I started by telling you about this problem that we only get to measure a subset of conditions and neurons, and this is a difficult problem, but it's something that we can approach with shuffle controls and other forms of cross-validation, and there's more work to be done to really characterize how this subsampling error causes distortions in the geometry. Trial-to-trial noise is a very important feature of biological neural systems, but it often gets ignored in this kind of analysis. And I gave you a flavor of one way of incorporating trial-to-trial noise by generalizing the Procrustes distance to kind of noisy point clouds or fuzzy point clouds rather than deterministic point cloud geometry. And then I said that the stochastic Procrustes distance can help us disentangle input-driven versus recurrently driven neural circuit dynamics, which I think is an interesting topic that seems like people in the audience are very interested in. So this is just one way that we can use trial-by-trial noise information to get at a neuroscientific question of interest. Okay, final thoughts, and then we'll have time for some questions. So comparative systems neuroscience, I hope I've convinced you, is a really fresh and exciting field. I think it's something that we're getting, it's a field where we have the experimental techniques that are needed to perform experiments at scale that are necessary to make these comparisons. So I think it's something that experimentalists can make really great contributions to the field by working in this direction. I also think it's an area that theorists can make a lot of contributions because the details of how you make these comparisons between systems, I hope I've convinced you, is a kind of subtle and important problem that we understand a lot of things already, but there's still things to be figured out and improved. And in that light, I talked about many different ways of approaching this problem of quantitatively measuring distances between neural systems. Some of these metrics are based on explicitly aligning neurons between systems. So for example, the Procrustes distance and variations of the Procrustes distance. Other methods that I told you about don't explicitly align the neural dimensions between systems. They're more based on implicitly aligning them somehow, for example, through specifying a decoding task or measuring pairwise distances within individual firing rate spaces. I'd argue that there's no best metric that you should always use to compare representations. Different metrics measure different things, and it's often useful to try out different metrics and see how they relate to each other. So I would argue that you should try different metrics and ultimately choose a metric that suits your scientific goal, the goal of your analysis. So for example, if you want to learn an explicit mapping between neural systems, if that's something that you're going to leverage for some other form of analysis in your paper, then an explicit mapping can be learned by methods like the Procrustes or soft matching distance. If you care less about mapping one animal onto another animal or all animals into a common space, but you instead care about some particular form of task information, I would think about developing a metric based on decoding task information. And if you want to be agnostic about what task information is important, you can fall back on methods like CKA or RSA, which, as I said in the second part of the talk, can just be thought of as the average decoding distance between systems averaged over many different tasks. And then finally, in this final part of the talk, I gave you a little bit of insight into how we think about comparing representations that are noisy or dynamical over time. I think that these are really hard problems, and you're going to have to look towards cutting edge methods in the literature, such as a dynamical similarity analysis, dynamical archetype analysis, or some of the methods that we've developed based on stochastic variance of the Procrustes distance. But there's a lot more work that needs to be done, I think, along this third line, and this is really something that theorists can make contributions to the field. And with that, I want to thank the organizers who helped administer this session at Cosyne, Leslie Weeks and Roxana. There's probably other people that I'm missing, but those are the two I interacted with. I want to thank the AV team. I want to thank Ava Dyer and Talmo Pereira, who were the chairs of the tutorial, of the Cosyne tutorial this year. Some of this work was supported by NIH funding and all of the TAs who helped prepare methods. So thanks. Okay, great. And we have 10 or so minutes for final questions if people have big picture questions or questions about the fourth part of the talk.
Hello. Thanks. This was really neat overall. I had a kind of specific question about the ending part just to make sure I got the intuition right. So your point about this, incorporating trial-to-trial variability, if I got it right, it boils down to, in the original Procrustes distance point, we're averaging across trials, but then your ultimate generalization was just, I'm going to concatenate all of my neuron time into one big vector and look at the mean and covariance of that?
Yes.
Okay,
yeah.
cool. As long as I got that, that's all I was looking for. Thanks.
Yeah, exactly. Yeah.
Thanks for your excellent tutorial. I have a small question about the Washington distance. I remember before we calculate the Washington distance, we should make sure to register 2-point clause. But when we combine the Procrustes distance and the Washington distance, yes, that figure. Yes.
Oh, yeah.
Maybe next.
I think I went back too far, but. Yeah, here, yeah.
Yes, here we first use the mean to calculate, to compute the...
No, sorry, yeah, so let me be specific about what we're doing here. So, so one problem with the Wasserstein distance is it actually suffers a curse of dimensionality. And if you only have a few number of trials per condition, we just limited ourselves to the mean and covariance of each condition. So, so first of all, the trial-by-trial variability is a mean and covariance around each condition. And then when we fit the rotational alignment here, we're minimizing the sum of squared Wasserstein distances between the point clouds. So the alignment here is not learned by the traditional Procrustes transformation. It's learned as the rotation that minimizes the Wasserstein distance rather than minimizes the distance between the means.
Okay.
I went over that too fast. A good question.
Okay.
Yeah.
I just want to make sure my understanding is correct. So it's not a step by step. So you compute the Washington distance and the practice distance jointly.
Yes. Yeah. So it turns out that there's unfortunately not a closed form solution for this optimization problem when you're optimizing over rotations and reflections in this case. But there's an iterative algorithm that at each iteration computes the Wasserstein distance and then finds a better rotation and reflection to align them iteratively.
Okay, thank you. Thank you very much.
Oh, yeah.
Oh, hi. I wanted to come back to the last point you made that there really is no best metric.
Yep.
And to see whether I kind of got the intuition, because actually even if you only focus on, let's say, this Procrustes distance,
yep.
There's an infinity of metrics that you can construct from it, right? Because
yeah, if you choose different pre-processing.
Exactly.
Yes, yeah.
Or because it involves taking the L2 norm, you could take the L2 norm with any semi-positive matrix.
Yeah, you could use a different. So the Procrustes distance uses like a least squares error criterion.
Exactly.
You could use a different error criterion.
And I guess the same applies also for the decoding distance. You could always, instead of taking the L2 norm, just put a semi-definite, like a posi-definite matrix in the middle. And this kind of raises the question, I mean, if you have, even for, let's say, a specific type of distance, an infinity of metrics that you can construct from it, why is the one that we are using, the kind of Euclidean one, more meaningful? And probably it is meaningful, you know?
Yeah, I mean, it keeps me up at night a little bit, to be honest. But I mean, I think for the specific example that you meant, like you can put a positive definite matrix in between, but then that would, I mean, I think the identity or a scaled identity matrix would be, in some sense, the most natural because you're fitting over rotations anyway, right? So you don't want to treat certain dimensions as more important than others, which is what you would do if you didn't have an identity matrix in the middle. So at least that particular class of infinity metrics, I might be able to rule out.
Yeah, and same then when you go to the probability space, when you go from the neuron state space, you make the transition to the space of conditions, and you look at distances between probability distributions. I guess there, the most natural one is the one from optimal transport, but you also mentioned another metric that you could use, this energy thing, I don't know it.
Yeah,
I guess there's also an infinity of metrics, like, you know, like just from a very mathematical point of view.
Yeah, so I think there's many different metrics, and I guess at the end of the day, you need to be a bit practically minded about it. So some of these metrics, the ones that we focused on, are easy to compute in most cases. So there is a computational tractability that goes into this, and you also just want to use standardized methods. So this is why again I think theorists can do a good service to the field by really characterizing what these different metrics care about and don't care about, and then if we can synthesize different, or if we can distill down the space of possible metrics into something that's easily digestible to end users, that's what I'm trying to work towards, and people in my group are trying to work towards. But it is a problem, and it's probably one that's never going to be fully solved. Yeah.
I had a question about the second part of your talk, about the equivalence between decoding-based RSA and CKA. Do we need to assume the kernel is linear in order to establish this equivalence, or we don't have to?
Yeah, so in the second part of the talk, I talked about RSA and CKA and their relation to linear decoding distances. This is using forms of CKA and RSA that are most popular based on the Euclidean pairwise distances or linear kernel in the case of CKA. There are nonlinear extensions of those methods, and those are quite interesting. You can think of them as comparing the topology sort of of the point clouds rather than the geometry. And you can also, I didn't say this, but you can interpret those variants of RSA and CKA. If you're careful, you can interpret them as a certain kind of nonlinear decoder, nonlinear decoding distance. Yeah, so great question.
Here. Thank you. I'm sorry. When we talk about mismatch system in the third part, we use a matrix to alignment these two systems, but in a tuning curve space. I mean, the dynamic, is in different period, the tuning curve of a neuron would be changed a lot in different test period.
Okay.
So it's possible that we use one matrix to align these two systems in the first period, but in the second period it would change. So that it makes sense.
Yeah. So okay. So I think what you're saying is that in certain neural experiments, you might record from neurons over a period of time, and then either at a later date or later in the session, the representations are either drifting or the animal is no longer paying as strong attention. And the system, or maybe some of the neurons are dropping out of the recording. There's various different ways that neural recordings can be non-stationary over time. None of the methods that I have talked about today, and I don't know of any work in this area that is really explicitly focused on how do you deal with comparisons with non-stationary data. You could imagine that you want to have methods that are robust to that and ignore the non-stationarity, or maybe you actually care about the non-stationarity if it's like an effect of attention or something like this. So I think you're pointing to an interesting direction, but I don't have too much intelligent to say about it yet. Thanks.
Thank you so much.
Hi, question here.
Yeah.
So once you have your alignment score, let's say you get an RSA score of 0.5.
Yep.
Can you say how special it is to be aligned to 0.5? Can you quantify the size of the solution space for different alignment scores?
So great. I'm going to go all the way back to the first part of the talk. So, I think what you're getting at, correct me if this is not your question, but I think what you're getting at is, at the end of the day, I can measure the distance between two systems, X and Y, and I'm going to get some number. And how do I interpret that number? Is that number meaningful at all? Like RSA of 0.5, does that mean 50% similar, whatever? One thing that I like about the metric space approach is if you have more than two systems, it becomes less about what is the absolute scale of the distances, but what are the relative distances across all of the systems you're comparing. So I went back to this slide because you can think of, here I have a 55 by 55 distance matrix that's computing distances between pairs of brain areas. And the absolute scale doesn't really matter too much. I can scale up all the distances by a factor of 10, and the patterning of the distances is preserved. So the multidimensional scaling will give me the same plot, even if the absolute scale of the distance is scaled up or down. So I don't know if I was answering the question you were getting at, but this is an important point of, is the thing that matters the actual distance score itself as a numerical value, or is the thing that matters the metric space that you're defining? And I'm kind of making a plea to think about this bigger picture of making comparisons across more than just two systems, but many at once. Did I answer your question?
Partially. I think the other part of my question is is my intuition correct that the lower the alignment score, the more ways there are to achieve that alignment score? And is there any way of quantifying that?
So I guess in terms of, I don't know about RSA and CKA specifically, but in terms of the Procrustes alignment distance, the Procrustes distance, so that is going to be, the closer that is to 0, the more similar the point clouds are. And the scale of the Procrustes distance, one of the things that's nice about that is that there is a very explicit interpretation of it in terms of root mean squared error and linear regression and predictivity. Sorry, it's way later than I realized in this slide deck. So I don't know. I feel like I'm maybe missing a little bit the point of your question, but we can.
Oh, let's take it offline.
You can ask after. Yeah.
Hello.
Yeah.
Thank you very much for your talk. I want to ask something that's a bit more macroscopic.
Yep.
So you started your talk by saying that we could apply such methods in the level of within organism or across same organism or eventually maybe across species.
Yep.
So I guess my question has mostly to do with this last part, which I think is very interesting.
Across species. Yep.
Across species. So I imagine that the information that's encoded in a certain neural network is different. So if we sample an n number of neurons in let's say a human, or let's say a monkey, or let's say a mouse, the information that we eventually can capture is quite different. So this subsampling issue becomes quite maybe bigger across organisms. And I wonder, in the end of the day, if you think we can figure out a way with which we can actually compare across species and get some informational component. I don't know if that's clear.
Yeah, so I mean, so is what you're saying is that I actually haven't done too much of these cross-species comparisons, but your intuition is that by these measures, they would actually be viewed as quite different. Like the individual neural responses look different.
I think potentially the information, in the way that information encoded, since also the sensory systems are different, everything is different across organisms, but eventually, potentially, the output goals should be same, maybe across the same sort of task between organisms.
I see.
I wonder how we could eventually align. This would be very interesting, I think, to be able to say that the output part is quite similar, even if the way that the information is encoded is different. But yeah, I guess it's more hard to do it across species.
Yeah, so I mean, I think one thing that we didn't really have time to cover is, and this maybe gets to the other question as well, is that once you compute a distance score between two animals and you find that the distance score is intermediate, let's say, like 50% alignment, you might be interested in digging into like what are the dimensions that are well aligned across the species and what are the dimensions that are maximally disaligned across species. So I think, I mean, that is actually quite possible with the Procrustes distance, if you think about it as fitting an alignment. Once I'm in this aligned space, I could look at conditions that are really still far apart and remain misaligned, and what are the conditions that actually get well matched. So yeah, I think that is a potential strength of using this particular form of distance metric, is that it learns an alignment between the systems, which then could maybe be used for other sorts of downstream analyses, where like I could do principal components analysis on the residuals of my Procrustes alignment and look for patterns within the residuals. Yeah.
Thank you very much.
Yeah.
Thank you for the tutorial. Before you introduce that you can calculate the representation and then align them, what's also common in neuroscience is that people will concatenate data and then perform PCA. I wonder what's the difference between these two different approaches.
Oh, interesting. They're probably going to end up being. Wait, I should be a little careful. There is a similar flavor, but I don't think there's going to be a direct correspondence there. I mean, one thing that you might worry about would be that the PCA would ignore one of the systems and preferentially just reconstruct from the other. But that would just give you a dimensionality reduction that can be applied to both systems, and it doesn't. It's not immediately obvious to me how to boil that into a metric for how similar or different the systems are. Good question. I hadn't thought about that before.
Thank you very much.
Go ahead.
Thank you for the really great talk. So earlier on, you were talking about how you think that in systems neuro, interanimal variability has kind of been underexplored, and I'm very sympathetic to that. And that got me thinking about how, well, if you explore that, it kind of depends on the task, right? You can apply these measures to compare representations across animals, depends on the task. And so my question is, do you think that we can use how similar different tasks make different animals to select between tasks? I mean, do we want more similarity across animals or less?
So maybe I'm inferring what your question is, but if I trained animals on many different tasks and then compared their similarity in their neural responses across those different tasks, to what extent is the animal-to-animal variability consistent or different across those different tasks? And it seems like to really get at that question, you almost want to define a distance metric over task space as well, which is something I think is really interesting. And I've talked with Leah Dunker a little bit about collaborating on defining similarity among different tasks. But yeah, I think that's a great question, and I don't have a great answer to it. Yeah. We're done?
I think we're done.
Thanks, everyone.